So once again, good morning or greetings of the day to all of you who have joined in. It's a pleasure to have all of you all here. So this is a good set of participants. Let me start off with a quick introduction about me. So I'm Ashwini Reddy. I'm a senior analytical training consultant with the SAS Institute, India. I'm based out of a city in India by the name of Mumbai, or otherwise like the Brits call it Bombay. So today we would be talking over a span of 4 hours in terms of how do you use analytics in the healthcare and the life sciences sector. And we are going to pick up a lot of demonstrations. We are going to pick up a lot of use cases relating to examples of either data management or analytics within healthcare and life sciences. So we are going to look at the entire process flow of the analytics life cycle, and it's going to be specific to the SAS usage. Now, since some of you all might possibly have questions, and there would be a flow that we would be following through, an agenda that we would be following through. After the introduction, I will introduce you to the agenda in order to understand what are the different topics that we are going to take up and how much time are we going to spend across those different topics. So before I could go ahead and do that, a quick reminder to all of you all to please rename yourselves in case if you have not used a correct, your full name rather, as your name, as your participant name on the Zoom participants list. Then please ensure that you use a full name, and probably in parentheses you can also specify in case if you're a student or if you belong to an academic institute or if you belong to the industry. And when I talk about the industry, you could either be working for a pharmaceutical organization, or you could be working for a clinical research organization, or even for that matter you could be working maybe for a healthcare administrative based company, or even in a hospital. Right, so that's a humble request to all of you out here. Please do ensure that you rename yourselves in the Zoom participant list according to your full name, and in parenthesis, just specify either, like I said earlier, either do you belong to the industry, or if you're from an academic institute, or if you're a student. That way, we get a feel as to who all have logged in today. And in case if there are certain questions coming in from your end, I can tailor make the answers to make it best suited towards your role, or I could also understand the perspective of your question. The second very important thing is request all the participants to please mute yourself till the time you don't have a doubt or a question. And the way we are going to go about with the questions is all of you do have access to the chat window, so you can just directly go ahead and plug in your questions in the chat window. As and when I see the questions coming up in the chat window, and as and when we do reach a logical break in our entire webinar session, that is when I will definitely take up your questions. So that's something that you can be rest assured of, right? So I do have another screen available with me. And on that second screen, I would be able to see all your questions that you would be plugging in. Now, some of y'all have been putting a message in the chat saying that not audible. Request you to just check your connections once again. So Revati, I would just suggest if you could, try to test your audio connections once. All right, so let's start off with the introduction to analytics for healthcare and life sciences specifically. Now, one of the very classic dialogue that you see out there, or even if you visit the SAS.com website, and if you get into the industry area of healthcare and life sciences, you do see a very classic dialogue, or a very classic or a catchy sentence that gets displayed on our website, which says that when it comes to the science of life, SAS cares. I mean, if you actually think about it, SAS being an analytical solution or an analytical platform, it is not specific to one particular industry of usage. And if you think about this history of SAS, the birth of SAS when it came into existence was primarily meant for hypothesis testing, or it was meant for statistical analysis in more specific areas, statistical analysis for life sciences and healthcare, right? So SAS came into existence because of a need or because of a requirement relating to the healthcare and life sciences. And over the years, over the decades, SAS has branched out to a lot of different industries out there. And having said that, SAS is one of the gold standards of a software which ends up being used for not just data analysis and reporting, but also from data management aspects. Um, Given that FDA has suggested as SAS to be one of the recommended softwares to be used for clinical trials data processing, right? So for very obvious reasons, the phrase is quite catchy, saying that when it comes to the science of life, SAS does care. And that's something that you would be able to see by a lot of customer success stories that have been, again, made available onto our website. I'm just highlighting a couple of them here. So you can always see this is a deck that I am going to share with you all towards the end of the session. More y'all can use it for your reference, just to get an understanding or a brief idea about SAS and the usage of SAS within within HLS. Yeah, so you do have a link here which is pointing out towards the industry page of life sciences. And if you're looking at some of the customer success stories, then you've got Parexel, which is one of the very large organizations out there that ends up using SAS software in AI applications, especially in terms of clinical development and healthcare. Now you've got a couple of more examples of the customer success stories. So you've got, for example, Oragonon, which is using again SAS for data analytics as well as visualizations and predictive modeling in order to understand the better patient outcomes. So that's one example of not a clinical trial specific use case. And then you also have Biomirics, where they end up using SAS in order to look at antimicrobial microbial resistance via data visualization. So this is something which is talked a lot recently. I don't know if any of you all do have a habit of reading newspapers every day in the morning, like the first thing that you wake up, given that nowadays you have social media, which is taking over even the news world. Traditionally what we end up you doing back in the day was you wake up in the morning, you would make yourself a cup of tea or a cup of coffee, and you would want to read the newspaper. But then nowadays you have the new age generation, which has showed us ways in terms of waking up in the morning and then directly going ahead and logging into Instagram or logging into Facebook or checking our WhatsApp messages from the previous night, or trying to top the list when it comes to a Snapchat and so on and so forth. Right. But then, in case if you are following up at any point in time during the day, even on the news, you would get to see a lot of discussion which is being initiated by the FDA or even by the World Health Organization in terms of antimicrobial resistance or the potency of antibiotics across different individuals due to an expanded usage of time being reduced over the years. Right, so Biomurics ends up looking at how can they fight against antimicrobial resistance via data visualization, and the data visualization can then be pushed into different areas such as computer vision, or it can also be put into image analysis. You can have fancy neural network models being used behind of it in order to get better analysis or quicker insights into what's happening as a ground reality. So if you look at these three customer success stories, they are spread across the entire industry of HLS in terms of looking at pharmaceutical trials or looking at clinical trials, looking at drug development. You're also looking at real world evidence data in terms of epidemiological studies or in terms of even multi-generational or just observational studies out there. Now, this is just three of the examples that I'm talking about here, and you can find a lot of other customer success stories published onto the SAS.com website. But to just put it in short, like a quick KPI or a quick view would be that you have about 100% of the life sciences companies in the Fortune 500 list that end up using SAS. So you can actually think about what kind of job requirements exist out there, at least in this Fortune 500 list of companies. Now, across that Fortune 500 list of companies, you have a number which is greater than 2,350 life sciences companies of all sizes that end up using SAS. So over and above the Fortune 500 list of companies, you also do have a lot of different brands under the healthcare and life sciences that end up using SAS for different purposes. And you're not just again looking at the focus on one geography, but then you have the usage of SAS or the presence of SAS, which is spread across 45 different countries. I mean, that's a very large number. And for a brand like SAS, which has always made its mark right from day one in the healthcare and the life sciences industry, is pretty obvious when you see a number like 45. So then you would be interested to look at what is it that SAS can do and what are the main areas of usage. So the very starting point and more prominence you do see in clinical research analytics, especially given that the FDA has mentioned that the SAS software is one of the gold standards or recommended softwares to be used in processing clinical trial data, right? So yeah, so clinical research analytics, that's one of the very specific areas that SAS ends up being used in. Then you have real world evidence and some of the very big brands, like, say, for example, AstraZeneca or GSK, they have extremely large teams that end up using SAS in order to look at or analyze epidemiological studies or observational studies and so on and so forth. Then it is also used in pharmaceutical manufacturing. So over and above just processing clinical trials data or analyzing clinical trials data, you can also enhance an operations aspect relating to the manufacturing of the different drugs or vaccines or therapeutic belonging to different therapeutic areas out there. And that's something that SAS can aid in with their entire ETS aspect or in terms of the optimization relating to operations research. And last but not the least, for obvious reasons, you cannot always expect a healthcare industry or a healthcare and life sciences industry to be not for profit. So when you start talking about patenting of drugs or when you talk about patenting of vaccinations, then there is definitely a commercial angle to it where you are definitely looking at a pharmaceutical brand trying to make a profit out of the investments that they have put into the clinical research or investments that they have put into developing that corresponding formulation, right? So how do they go about with the analysis of the commercial aspects of it? That's also another area where you can definitely use SAS. Now, all of these different use cases or all of these different areas are achieved with three major buckets of the SAS software. One relating to artificial intelligence. So you have a lot of uh, inbuilt point and click predictive modeling, machine learning modeling techniques available onto the SAS platform. So examples would be the large language models that end up being used in text analytics. Another example would be the neural network modeling that ends up being used in computer vision. Then you can also talk about SVM, which is your support vector machines. These are certain predictive modeling techniques or machine learning techniques that aid in artificial intelligence. Therefore, that can very easily be built and analyzed upon your data with the help of just simple point and click tools instead of just going ahead and trying to use voluminous amounts of code. The second important block would be, given that now we are looking at the volume of data increasing much in much larger density or in much larger speed as compared to what you had it probably even a decade back, then investing in an on-site infrastructure in terms of having storage capacities or in having a very large architecture that could allow you to have parallel processing or quick processing of information. In order to avoid those stoppages, you then have cloud computing, which is also available. And we do have our official partners, such as Microsoft, that allows us to host our applications or our solutions onto the cloud. And the third very important block to all of this, especially when it comes to real world evidence data, is relating to the Internet of Things analytics for medical devices. So some of our applications or some of our tools, which I would also be talking about a little bit in like more like a high level view overview. Towards the end of our webinar would be relating to a tool known as a SAS event stream processing that can be used in order to uh, get in live feed of data from different queues, right? So these are the three building blocks of our SaaS platform, which allow a lot of brands out there in order to achieve a success in terms of using SaaS for healthcare and life sciences based analytics. Now, you must be wondering that, okay, when you talk about all of these different aspects, fine, they sound wonderful, they sound very pink of health, and they sound like, okay, you know what, these brands are doing really good for themselves, and SaaS is enabling them in order to do it. But then you would want to start thinking about saying that, okay, hey, listen, I have studied analytics, and I do know that there are different phases or there are different steps that I need to follow in order to meet or understand or derive a solution or an insight for a particular business requirement. Now, this is something which is very generic across all industries. It is not something which is going to be very specific to the healthcare and life sciences per se. So you would then want to start thinking about, okay, those 7 steps that I do know or that I have studied in theory, or when I initiated my career in analytics, my managers or my project group seniors, they ended up talking about the different 7 steps. And those different 7 steps would first start off with trying to ask, what is the problem? So you would want to try and identify, are you looking at, say, for example, if you're talking very specifically to drug development, are you trying to figure out the efficacy of the new formulation that you have designed or developed? If you're talking about epidemiological studies, or if you're talking about epidemics or pandemics, rather, given that all of us here do know how COVID spread. So you have a very classic factor there known as a reproduction factor, the reproduction rate, which decides whether, which is also calculated on the basis of the incidence, prevalence, and certain other demographical aspects. So you would want to try and look at, okay, what is the reproduction rate of a condition or a illness or an epidemic out there? So the starting point is always where you would end up asking, what is the problem that you have, or what is it that you are trying to derive an insight for? And on the basis of that, you start then looking at data points. You look at from what could be a different sources of information. Now, again, not all information in healthcare and life sciences would be available under one roof. There would be information that you might want to pick up from WHO. There would be information that you would also want to pick up from your World Bank, because there could be a possibility of macroeconomic factors also having an influence on the overall health of a community or the overall health of a particular country, right? So you would end up looking at collaborating data from different roofs and not just one roof, which would be only your organization setup. Then you would want to try and explore the data. And on the basis of the exploration, you would want to try and figure out saying that, okay, should I be looking at clubbing information together, or should I be looking at, translating my tables maybe from a horizontal to a vertical structure or vice versa? Are there any data quality issues that I should be fixing before I go ahead and use it? Missingness of information could be a huge uh, problem when you talk about healthcare and life sciences. So then how do you deal with that missingness of information? So these are certain things that you start thinking about once when you do have the different data points that you should be trying to analyze in order to answer a question that you have asked or that your business team has asked you. You would then want to move ahead to the modeling part. And when you talk about the modeling part, again, you're not limited to just using the traditional predictive modeling parts, but then you would also want to look at, hey, why not? If I have a retail industry who's going to go ahead and build an analysis with the help of neural networks, or if I'm going to have the financial industry, which is going to use logistic regression, why should I always be limiting my safety or my efficacy analysis in HLS to just parametric testing or non parametric testing or the classical survival analysis? Why can I not take it forward one step to look at certain more complicated predictive modeling techniques, right? So you can always, again, the learning from another industry can always be brought into the HLS, and you know again, it would all depend upon whether you are asking the right questions and whether those modeling requirements would fit your need then. And then once when you have your analysis in place, once when you know that you can have some kind of a predictions coming out of your modeling systems, you then go ahead and try to implement them. You then try to take certain actions against the results, and then you go ahead and perform an evaluation over an expanded period of time, right? So the classic example would be when COVID hit us in 2020, early 2020, and lasted about a year, where it was quite severe. The reproduction rate back then would have been a much higher value, and on the basis of what actions had to be taken, uh, the each geography would have acted in a very different manner that could have been very specific to their demographics, right? But then over the years, you have did notice that the reproduction rate of the COVID cases have reduced to a very large extent, for obvious reasons, that a lot of the incidences or a lot of the prevalences were curtailed, they were treated, and then there was a sudden drop in the number of people who started contracting COVID. So then the way the government acts at that point in time, you cannot use a same model that you would have used as a part of the 2020 in 2024 or in 2023 for that matter, right? So you need to have a continuous evaluation process, and the evaluation process is not just going to work in terms of looking at your data changes, but it's also going to take into consideration your macro as well as the microeconomic factors. And hence I made a point saying that when you ask a particular question, not necessary that the data points that you're going to then collaborate or work with is all the data that you're just going to pick up under one roof, but then it is going to be data coming across from multiple different areas. So this entire 7-step process, except for uh, where you manually have to bring in the data, keeping that part out. But then if you have to look at the more technical aspects of the analytics lifecycle, then that can be very easily mapped to the 3-step approach just under one web browser, or pretty much under one solution set, in order to give you a complete end-to-end workflow, right? So when you talk about asking questions or preparing your data, you have a couple of applications that can be used in order to work with the data. When you talk about exploring your data and using certain predictive modeling techniques or statistical modeling techniques, rather, that comes under the discovery area. And then subsequently, when you talk about implementing, acting, or evaluating, or rather governance, you then look at the entire deployment phase. So in technical terms, when you talk about the SAS umbrella, you have the entire 7-step process that is very beautifully clubbed into just 3 areas of work: one relating to the data, one relating to discovery, and one relating to deployment. So if I have to just show you here, one of our uh, more recent platforms, which is known as the SAS Wire platform. The SAS Wire platform is, again, it doesn't require you to have any desktop installations. All that you need is just a web browser. So here you can see that I am connected to the SAS solutions or to the SAS tools. And this is a server which has been hosted for us, but I do not need to have any kind of installations onto my computer. All that I need is maybe a Chrome or a Microsoft Edge browser. And if I just click on this cute little hamburger icon on the left hand top side corner, then here you do notice that I can work with the data within this application. I can go ahead and perform discovery analysis, such as exploring my data, building predictive models that could come under exploration and visualizing the data, building the models. Then I can also go ahead and um, act on my models. I can manage my models, I can govern, I can evaluate my model performance with the help of managing the models and building the decisions. over and above this, if I have to just look at all the content, I do have something like a OneDrive, or rather a similar OneDrive application, which is known as a Share and Collaborate. It's known as a SAS drive. And over and above this, if there is anything that I cannot do with point and click, all these applications that you see here under the analytics lifecycle are all point and click applications. If there's anything that I cannot do with a point and click feature, I can also go ahead and develop my own code. Right, so I do have an option to get into SAS Studio, and then I can work with my SAS programming language in order to make certain quick analysis. Right, so each of these applications that come under the analytics lifecycle onto the SAS via the new platform, it does not require you to have any installation specifically to each of these tools. And all that you need is just one web browser, and you can navigate through the different aspects of the different areas, or rather the three phases of the analytics lifecycle in one single go. Right? So, sorry, let me just come back here. Yeah. So this is what I was trying to show you with the help of just that one quick view, which was the Wire platform. And it has a singular focus on the entire analytics life cycle, all the three steps, or rather all the three phases. Sorry, I request all the participants to please mute themselves. Okay, thank you. All right, yeah, sorry for that. So the base being the analytics life cycle very well integrates with the SAS platform. And when you talk about the SAS Wire platform, like I showed you, me as a user, I just need to have a web browser, and then I can very cutely navigate inside using the different tools that I have with me. If you're talking about the SAS 9, which was our traditional on-site, more of a desktop-based usage or desktop-based license, that ended up looking at having different applications. Some of them needed you to have an installation on your computer, but the SAS 9 platform also very well integrates with the analytics lifecycle. And with the help of this entire analytics lifecycle, whatever outputs, whatever insights you end up getting, you can use that as one of the very key features within the capabilities and solutions that SAS also offers. So across different industries, like say in case if you're talking about banking and finance, then you have certain very classic softwares that are being used out there. So you have our SAS risk solution, risk management solution, which looks at current expected credit loss. It looks at IFRS 9, IFRS 17. It also, then you have the SAS anti-money laundering solution, which looks at fraud detection. Then when you talk about healthcare and life sciences, and you've got certain applications or certain solutions known as your cohort builders that helps you in designing the uh, sample of your clinical trial. Then you have the life sciences analytics framework that can be used for real world evidence. And if you're looking at retail, then you've got something like customer intelligence 360 that can be used in order to get a complete demographic information, or can also be used as a marketing tool within the retail industry, right? So that there are different capabilities, there are different solutions that have been built over and above the tools that allow you to subsequently work with the three important parts of the analytics lifecycle, right? So when you're talking about analytics for healthcare and life sciences, these are just certain quick bullet points in terms of what is it that you can do when you talk about accessing the data, what is it that you can do when you talk about preparing the data. You're then looking at visualizing, exploring the data, and then subsequently the statistical analysis part. So these are just a few bullet points. So when I share this deck with all of you later on, so I will be sharing this deck with y'all. Y'all can just have a look at these points in case if you just want to get a quick view in terms of what are the different things that SAS can do across the three breakups of data discovery and subsequently deployment or statistical analysis. All right, so I'm going to take a pause here to check if any of you do have any questions. Okay, so I have a couple of questions coming in here. Will the session be recorded for us to view later as well? So the session is definitely being recorded. I would just have to check and get back to y'all as to whether the recording would be shared with y'all for later. So let me, try to figure that out towards the end of the session, and I shall again address your question at that point in time. Hope the slides will be shared later. So we are going to send out a PDF copy of this deck file because it has a complete information about SAS in healthcare and life sciences and certain examples of our demonstration. So I would be sharing a PDF copy of this deck file that we are going to use. Right. Any other questions? Just a quick check. Do any of y'all have any questions? Do y'all want to check about the basic information that we have spoken about, like just the introduction part as to how SAS can work with the healthcare and the life sciences industry? All right. So if there are no questions, the agenda that we are going to look at for the next good 3 hours is we are going to try and focus about how SAS cares and what is it that we've spoken about. A lot of things we've spoken about, the different applications under the data phase. Spoken about a couple of applications under the exploratory phase or the discovery phase, and then subsequently the deployment phase, right? So we would also want to try and see, if not at least for the deployment part, the first entire analytics life cycle, which is preparing the data to building a particular statistical model. How do we go about and do the same activity onto the SAS platform, right? So we are going to go ahead and pick up examples relating to exploring the data, accessing the data. How do you go about and prepare it? Some bits here and there. We are going to use SAS programming examples, and then we are also going to go ahead and move on to the more of the point and click applications. So in today's session, we are not just going to limit ourselves to look at the Wire platform because some of you all might be very familiar with the 9X platform as well. So some of the examples we're going to take up on the 9X platform, and some examples we are definitely also going to take up on the Wire platform. And the reason why we are going to do that is, to show you that do not be afraid in case if you're planning to move on to the Wire platform. Do not be afraid of doing so because pretty much of what you used to do at one point in time becomes quite easy, in fact, when you start doing it onto the Wire platform. Right, so the agenda for today, the first 45 minutes starting from 9:15, we are going to take like a quick 5 minutes refresher break. Following this introduction, we are going to start off with preparing our data from 9:15 going up to about 10. So that's like about 45 minutes. Now this time is in the Indian Standard Time. So I would request the participants from all other countries to just work on the basis of the duration. Right. After we speak about preparing your data, we are going to move on for a nice break of 15 to 20 minutes break. And then following that, we are going to look at how do you go ahead and visualize the data. We are also going to take up certain examples, two rather very cute examples when it comes to hypothesis testing. And uh, the reason why we are going to talk about hypothesis testing here today is because that becomes a starting point for your analysis when it comes to healthcare and life sciences, right? So especially when you talk about clinical trials, hypothesis testing is a must do. So that's something that we are going to talk about. Then we have a dedicated 15 minutes for a Q&A. In case if you do not have any questions during that 15 minutes, then you can also consider it like a break. And also this part of 15 minutes has been kept as a buffer in case if we do run out of time, then I can steal a couple of minutes from there. Finally, we are going to talk about the predictive modeling techniques. And here I'm going to show you all with two very interesting examples, one relating to decision trees. So we are going to quickly look at how the Wire platform has an application, or rather has a tool known as a model studio, and how the model studio can be used to build decision trees. And when I talk about decision trees, it's not just a simple decision tree, but then we can also look at gradient boosting. You can also look at forest And then subsequently, you would always be interested in the scoring process, or you would also be interested in the model comparison, right? So we already have a predefined demonstration, but I'm just going to walk you through how easy it is to work with it. And the last 15 minutes would be a wrap up, which is going to talk about some of the solutions relating or specific to the healthcare and life sciences area, or social and behavioral research, which is also very important for our industry. And we are then going to wrap up the session with me introducing certain contact details to y'all, that you all can reach out to in case if you would be interested in signing up for certain education based activities, or if you are interested to gain more information relating to the SAS analytics for HLS. Now, across this entire agenda, these are the three main examples that we are going to take up. So we are going to look at programming examples. We are going to look at, following that, we are going to look at two tools onto the VIA platform. One is SAS Visual Analytics, which is used from a BI perspective, which is, the dashboarding or the reporting perspective. And then we are going to look at another tool, which is known as the SAS Visual Data Mining and Machine Learning, which is primarily used for predictive modeling, machine learning, and it is also used for uh, visual text analytics. So, right, so two of those examples are something that we're going to take up here, like I said earlier, one relating to decision trees, and the second one relating to text analytics. Because again, like all of us do know here, when you talk about healthcare data, it can be really messy or really cluttery or dirty in nature. Okay, so here we are going to look at a serious example. And the reason why I am taking this example is because, like I said earlier, you have four main areas of use cases when it comes to SAS analytics in the entire healthcare and the life sciences sector, right? So you're looking at clinical research analytics, you're looking at real world evidence, pharmaceutical manufacturing, and pharmaceutical commercial analytics. And one of the starting points of the usage of SAS is in terms of preparing your data with in a very specific format that is required by the FDA. Now, most of the drugs that end up being developed out there, drugs, vaccines, biologics, or any other consumables such as devices. So if you're looking at any of these different interventions that need to be approved by the FDA body, then the clinical trial for which you are collecting all of that information, or during the course of the clinical trial, all the information that you end up collecting, which can be relating to the patient, it can be relating to the treatments or the interventions, it can be relating to any kind of problems that your patient suffered during the course of your clinical trial. And it can also be relating to certain extreme um, adverse events that happen to a patient, such as paralysis or requirements of hospitalizations or ambulatory facilities or death, even for that matter. So, when you look at all of the different happenings, interventions, performances in terms of safety or efficacy of your intervention on the human body, you would also want to collect all of this information because it acts as evidence, and this evidence then needs to go ahead and get submitted and reviewed by the two very important bodies under the FDA. So when you're looking at a drug trial, then you have the Center for Drug Evaluation and Research. And when you're looking at biologics evaluation, then you have the CBER, which stands for the Center for Biologics Evaluation and Research. Now, these two bodies of reviewers are not going to review your data if your data is not in a harmonized format. So a very classic example of this would be, let's take up an example here. So when I have to just look as gender as a variable, and here I am not talking about the social gender. Let me just correct myself. I'm not talking about social gender here, but I'm talking very much about biological sex, because the moment you talk about medical sciences, it's the first preference goes to your biological sex. And then, for obvious reasons, your psychological well-being or the psychological impact of how you perceive yourself in terms of gender, that is also another area of interest. But here I'm just talking about gender as in your biological sex and not the social gender. So for each and every clinical trial which is conducted on human patients out there, one of the very important piece of information that you end up collecting about your patients is the sex. right? And traditionally, when you talk about biological sex, it can be only three values. It can either be a female, the human being can be, or the patient can be a male, or the person can be an undifferentiated gender, right? So if you have both the female and the male chromosomes present in the human body, then obviously you become undifferentiated. So if I have to record this information, and if I belong to pharmaceutical company 1 and you belong to pharmaceutical company 2, there's another person who belongs to pharmaceutical company 3, what would then happen is I could have recorded female as female, you could have recorded female as F, and then I could have had a third person who would have recorded female as a number. So this data is non-standard, and then it becomes very difficult for a reviewer to pull out that much amount of time and first study the data dictionaries of all the three different pharmaceutical organizations, correct, who are conducting a clinical trial. Now, in order to save up on that, the FDA adopted the guidelines of data harmonization from a not for profit organization known as the CDISC. CDISC stands for the Clinical Data Interchange Standards Consortium. And it looks at very specific geographies. So, geographies could be, um, you know, some of the countries in Asia. Then you can have Australia, India, Sri Lanka, uh, Thailand. So all of these different places, US for obvious reasons, UK as well comes into the picture. Not all the geographies, but very specific geographies in terms of countries that end up having pharmaceutical companies that are going to develop drugs for over the borders and needs to get an approval from the FDA would have a requirement of standardizing the data according to the logic that has been written down in the guidelines of the CDISC body. And the CDISC body has come out with two very specific guidelines, one relating to the SCTM that stands for the study data tabulation model. The study data tabulation model looks at capturing all the necessary information. Please note it's not a guideline in order for you to collect information during the course of a clinical trial. But once when you have collected the necessary information for a particular clinical trial, you have to very specifically design it in a standard or a harmonized format. Right? So the SETM looks at picking up all of that raw data, tabulating it in a very standard manner, and then you're looking at the second set of the guideline, which is the analysis data set model. Now, this analysis data set model is going to help you to create analytical base tables, which would be a combination of data from the different SCTM tables in order to answer the two very important goals of pretty much any clinical trial and experimental clinical trial out there, relating to safety as your primary goal and your secondary goal being efficacy. And the moment you talk about efficacy, you do have a lot of different endpoints that get divided into primary endpoints and secondary endpoints. Right, so this that you see on the left right hand side of my screen looks at dividing all the data that has been collected during the course of a clinical trial into very demographic based information. Following that, any kind of interventions or rather any kind of substances that have, sorry, not substances, but any kind of consumables that have been given to the patient, either relating to the study or in order to take care of certain other problems that the patient is suffering from, that gets clubbed under interventions. You then have events that's going to record any kind of information relating to possible side effects or possible problems that the patients are suffering from. Then you've got findings that's going to record all the information relating to different tests or physical examinations that have been conducted for our patients, right? So some of the examples that you can see here under the findings, I've just written laboratory test results. But then if you think about it, you've got physical examinations, you've got vital signs, you've got simple questionnaires, and a lot of different performance-based tests that can be used in efficacy analysis would all reside under very specific domains or very specific tables within the findings general observation class. And over and above this, the SCTM is not going to limit you to working with only the data that they expect it from you. right? But then if you have some data that you have collected, which is going to be very specific to your clinical trial, but unfortunately it might not be listed down in the guidelines, then you also do have an option to create your own domains, right? But then all of these different domains that you create under these different classes as well as these special purpose domains need to follow a very specific structure. So if I just take up the example, here you see that this is a case report form of how patient information would be collected at the start of my clinical trial. So once when I get the information consent form signed from my patient, that is when I can initiate even the collection of patient information. It would be unethical for me to even pick up patient information prior to signing the information consent form, right? So the patient information ends up being recorded at the very start, which would be at a screening phase of my clinical trial. And it's usually recorded on pen and paper, or nowadays you might have fancy devices that could record it onto a screen, a form, they're in a digital form, also known as electronic case report form, and then that can be directly pushed into a database at the backend, right? So if I just look at some of the information that has been captured here, we've got the birth date, the age units. Now, if I'm looking at a clinical trial, I can conduct clinical trials for different conditions, for different therapeutic areas. And not always I would end up looking at adults, right? There could be a possibility where certain clinical trials are looking at neonatal conditions or conditions of infancy, sorry, conditions during the stages of infancy or childhood, right? So that time, I might not always want to record age as years. There could be a possibility where I might end up recording age as days, or I might also end up recording age as months. And hence, there is a bifurcation in pretty much most of your case report forms associated with different clinical trials in terms of age being one area and age units being another question altogether. You then have the biological sex, and then you also do have certain other demographic factors such as the ethnicity and the race, which ends up being captured during the course of your clinical trial. Now, the reason why, again, you would want to capture this ethnicity or you would want to capture the race information is because each and every race, or rather each and every culture, has a very specific habit, right? Or it has a uniqueness associated to it. So classic example would be, for those of y'all who are in India, you might be familiar with this, but those of y'all who are logged in from the other parts of the world, just hear me out in this case. So when you talk about India as a country, and if I have to divide it into just two halves, which is the north of India and the south of India, the dietary patterns across these two different halves are very different. So the south of India is more of a heavy rice eater. And when you talk about north of India tends to be more of a heavy gluten or gluten based products such as wheat, jawar, bajra, and so on and so forth. So you end up having more of, uh, let's say, wheat as an example, being consumed in the north of India as compared to the south of India. Now, the way this works, as an example, for a diabetic patient, would be very different. So, a person who suffers from diabetes or who has a diabetes as a lifestyle condition from the north of India, the doctors would end up suggesting that you have a balanced diet where you cut down on your wheat intake and you increase a little bit of your rice intake, right? So, you would want to have that balance established between the two. But that same suggestion would not be given to a person from the south of India because the south of India person is anyway consuming a lot of rice. So the suggestion is then reversed around where the doctor would end up suggesting, saying that, hey, listen, bring down your rice intake and maybe start introducing a little bit more gluten in your diet, right? So the balance is achieved nonetheless across both the suggestions. However, the suggestions are more tailor made towards the dietary habits or towards the dietary patterns that have been influenced because of two very important aspects: cultural uh, impacts as well as geographic impacts, right? So these two playing major roles are nonetheless also going to impact two very important aspects: how an intervention works in the human body and how the human body is going to react towards a particular intervention, right? So Having said that, that comes under a very technical area known as pharmacokinetics and pharmacodynamics. And in order to also study those aspects, you would want to also bring in certain demographic factors such as the ethnicity or the race details, along with the geography information as well. Now, all of this information would end up being captured in very raw format, and it might be very difficult for us to use. So let's jump into our example here. So I'm just going to come back, and we are going to take up, like a programming example in this case. And before I could go ahead and do that, let me just also show you the raw data. Okay. All right, so I have already created, so one of the ways that you can access data on SAS is via a concept known as libraries. So I already have, and libraries just point out to a physical location as to where your data is stored. So it's more like a house name to your name, right? So it's something of the kind. So if I just expand libraries, and here I'm just going to come down to a library by the name of raw. And if I expand this for an example clinical trial that I'm looking at, I have all my patient information in terms of description of my patients captured into a data set by the name of demographics. So I'm just going to go ahead and open up this data set. And let's have a look at the kind of information which is there inside. Now, there are a lot of variables here. Now, first, if I have to just, if for the first time ever, me as a programmer who belongs to the same pharmaceutical company is looking at this data, in the first go, I understand very few variables. So I understand subject, I understand gender by the way it is written, I understand race. Very classic DOB would stand for date of birth. So these are certain variables just off the hat or without looking at the data. I can just understand saying that, okay, gender might capture the biological sex. Race might have information like in terms of whether the person is black, white, or Asian, African, and so on and so forth. When I come down here, if I look at DOB, then I know that, okay, the patient's date of birth is what is going to be captured in this particular column. But over and above this, there are certain other variables that I don't understand just by looking at the variable name. Second, when I start looking at the data, here I do notice that there are 60 observations in or 60 rows in this corresponding data set. I can draw an interpretation saying that maybe these 60 observations are pointing out to 60 subjects that have been enrolled onto my clinical trial. But when I start preparing my data, I would first have to check whether I do have 60 patients or do I have any kind of a duplicacy in terms of the subject numbers, right? Following this, for the variable that I did understand, like in terms of gender, I then start looking at the data and I figure out that, hey, gender can be seen as 1 and 2. Now I'm in a fix. Me being an in-house programmer, me being an in-house person for this particular clinical trial, I myself, I do not understand what is 1 pointing out to, what is 2 pointing out to in the first go. Which brings me to the point that if the CDER, which is the Center for Drug Evaluation and Research, if they have to look at this data, they would also not be able to understand, is subject 101 a female, or is subject 101 a male, or is subject 101 an undifferentiated sex, right? The same thing goes even with the race. I do not get a lot of information, me as a user, and hence for obvious reasons, even my regulatory authorities are not going to figure this out in the first go. You're then looking at the pain point that I spoke about earlier, where you would be expecting a regulatory authority to put in time in order to read the data dictionaries to answer this corresponding question, right? So me as a programmer, in case if I have to start looking at the raw data and translate it according to the CDISC guidelines in very specific formats, I can just quickly first try and look at an overall information of the data within. So I can just run a condensed procedure that's just going to give me a basic description of the data, point it out towards the library in which my data is stored and hence the data set. And I can just go ahead and plug in the run statement and execute it. So SAS being a fourth generation language, the way you talk on a day-to-day basis, I mean, a little more technical language that you use when you talk is something that you also type it down. So it's not as complicated as any of the object-oriented programming that possibly you would have heard of under C or C++ or Java and so on and so forth. Right, so here I do get a list of variables, and then for each of the variable name, I can see the label. So, okay, so I do get certain understanding now for some of the other columns, like say, for example, IC date stands for information consent date. So I know that, okay, a patient would have signed the information consent form on a particular date. Then I have the last date of contact. I have randomization date. So this is pointing out towards my clinical trial being a randomized clinical trial where the patient assignments to the different treatment groups happen on a random basis or a random logic rather than a manual assignment, right? So this is something that I understand now. Again, this is something that I'm not going to push it across through the CDISC bodies. So the next thing I would want to start thinking about is that, okay, having said that CDISC exists and CDISC has two guidelines under it, and I have collected some data, now what do I do next? What kind of variables do I create? So this is an example of a demographics domain, which comes under special purpose domain models. And here you do notice that there are very specific variable names that need to be followed. The type of these variables also have to be very specific, right? So notice this, most of the variables are a character data type, and the only variable which is numeric is an age variable. Now, each of these variables have a role and also have a core expected to it. So you can see that there are two very specific cores that are written down here. One is required, one is expected. Required means that it's a compulsory submission. You need to have values. You cannot have missingness of information and so on and so forth. And the moment you talk about a core of expected, then okay, you need to include that variable in your submission, but under certain situations, there could be a possibility where you might not have information for that corresponding variable. Classic example would be, just have a look at this variable, which is DTH Now, I have not noted down the expansion here, but then if I have to tell you what would this variable mean, this variable would end up capturing the date and time on which a patient has died during the course of my clinical trial, right? Now, for obvious reasons, if a patient has not died during the course of my clinical trial, this variable is going to carry a blank value, a missing value, or rather a not applicable value, correct? But if a patient has died, then I would have to specify the date and the time of death for that corresponding patient, correct? Now, in such a situation, you cannot force, nobody can force you to give a death date value for patients who are still surviving, and hence the core ends up being expected rather than required. Having said that, when you start looking at the creation of these different variables, there would be certain terms and conditions that you would also have to keep in mind. Like an example would be the moment you talk about the creation of the variables, each of these variables have to follow a version formatting, which is known as a SAS version 5. Now, this holds true as per the end of 2023. Starting from 2024, there are certain changes that are taking place in terms of making the variable names a bit more compatible to new age generation systems, right? Well, back in the day, you had a lot of uh, limitations across cross database functionality. So there used to be multiple databases or multiple source systems out there that would not allow you to have variable names beyond 8 characters in length, or that could not have variable values beyond 200 characters in length. And hence, the SAS version 5 also ended up following through the same restrictions and thereby got adopted by the CDISC body. This is about to change, and the changes would end up being published sooner within the 2024 year itself. So it's more as work in progress at their end. So yeah, so as of now, we're going to talk about the SAS version 5. So you see that the character variable length would end up having a restriction of 200 characters until and unless necessary. You would want to use the entire 200 characters if you're looking at a large string of information like comments or doctor notes. But then if you just have simple information like a sex or a treatment name, then you would want to use the stipulated number of characters therein. Then your variable names also have to follow a very specific formatting. The variable labels also should be within 40 characters in length itself. And a very interesting part is any kind of a date or time character variable has to be represented as a character data type in a very specific format of the ISO 8601. And it needs to appear like a 4-digit year hyphen a two digit month, a 2-digit day, followed by a T separator, the hours, the minutes, and the seconds portion. Now, when you're looking at this entire understanding of the CDIS guidelines, yes, first our theoretical knowledge of the guidelines and what kind of a variables have to get into what kind of a domain is very important. That becomes a starting point for us. And once when we have that necessary information, we then have to start looking at translating all of this data into um, a specific look and feel, right? So I already have that data written for me. And this is something that we also teach in our Clinical Trials Academy, Programming Academy. So traditionally in the industry, you end up looking at a 5-step process the moment you talk about preparing your clinical trial data for submission. So the very first step is, given that the SCTM domains that you end up creating need to follow a very important structure, you can just go ahead and create a shell that can hold the definition of all of those variables. So here you do notice that with the help of a lens statement as a part of the step 1, with the help of a lens statement, I am creating a lot of different variables. I am giving labels for these variables. Now, each of the variable is either defined as a character according to the guidelines, and you will also notice that the length of the variables is nowhere exceeding the 200 character restriction. Following that, when you talk about the labels, if you also look at the labels, you will notice that the labels do not exceed 40 characters. So I can just create first a shell that would just have the structure of the submission data set that I'm interested to create. Next, I would want to identify from where should I be picking up this data. And I identify that I end up picking up this data from my clinical trial library, which is raw. I have a data set by the name of demographic. Next part would be to then look at how would you would want to submit all of this information. So like I said earlier, there are certain variables in our input data set that are numeric here. So you can see gender, which is your biological sex, or the race is a numeric value. Now these numeric values are not valid for the submission, because if I have to look at these two variables in my guidelines, then my guidelines clearly tell me that a variable such as sex has to be character in nature, and a variable such as race also has to be character in nature, which means that, again, there would be a specific list of values that I need to submit, right? So I would want to then create certain formats as well by understanding what is my numeric values pointing out to, what CDISC wants, and I perform a mapping between the two in order to get the values recorded. So for example, sex, we saw values like 1 and 2 in our data. So CDISC needs the values to be submitted as for males, you're looking at M, for females, you're looking at F. So that's the values that you can see here. Any kind of missing values, is this U is pointing out towards unknown, undifferentiated would end up being written down as undifferentiated itself. Now, when you talk about race, you're looking at the values of 1, 2, 3, which points out to white, black or African American, 3 stands out for Asian, and these are all control lists. These are all standard values according to our guidelines. That's something that I cannot change. And even the way I record this information from the source, I can record information as male or female, right? So here, if you see sex, you have the information coming in as female, male, unknown, undifferentiated. However, I do not submit the values this way. I would end up submitting the values like F, M, U, or undifferentiated. Right? So that mapping or that building of the gaps between the way you have collected your raw data and what is the standard submissions under the CDIS guidelines, that's something that you end up doing as a part of step 2 or step 3, which is mapping your data, your source data to the submission data set. Following that, if there are any kind of variables that you would want to calculate, that's something that you would want to do. So certain identifier variables, if you're looking at a multi-center trial, then you would need to specify a site ID. That becomes a very important identifier in this corresponding domain. So you define your site IDs according to a particular logic. And the two most important parts are to uh, identify the treatment start date for a patient and the treatment end date for a patient, because that would also be an indication towards whether your patients are successfully completing the clinical trial or not. Right, so that's something that ends up being captured across. You start calculating it with the help of maybe the same input data set or a different data set. Here we have a separate data set that is being used, such as a dosing data set, and that data set ends up being used to calculate your first dose date and your last dose date. Now, this first dose date and your last dose date subsequently in the code is then being used in order to reference towards the overall treatment start date for a particular patient, overall treatment end date for a particular patient, and the overall clinical trial start date for a patient and clinical trial end date for a patient. Now, the reason why here the same variables have been used is there could be a possibility where there's not a lot of uh, follow-up periods being plugged into place, or there's no preparatory period that's been plugged into place, and the clinical trial is looking at a duration where you enroll the patient, you screen the patient for the inclusion criteria, you randomize the patient, and then you go ahead and you start with the treatment. Right, so if it's a pretty sorted, simple clinical trial, then your clinical trial start date, end dates, and the treatment start dates, end dates would end up pointing out towards the pretty much same information from your source. Right, so in this programming example, you do notice that there are a lot of different steps that are being used. Some places we have ended up using a data step. Some places you also do notice that there is an SQL code which comes into the picture. And once when you go through all of these different steps, I mean, once when you process your raw data, you then end up looking at the final step where you would want to uh, capture all of this raw data into your output or your submission domain. Right, so in that case, the structure which you would have created in the very first step is used as a structure in your last step, and all the data subsequently would end up coming from something like a flat file where you have already combined information from your demographics and you have combined information from the dosing. Correct. And with the help of just simple assignment statements, now this is easy for those of you all to understand who do come with a programming background. For the rest of you all, just get an idea saying that, okay, this is how your data is prepared. You are looking at about 150 to 200 lines of code to just process one simple domain submission, right? So then you end up looking at the usage of different assignment statements, and whatever those control lists or code lists that you would have created, you would want to attach them to the raw variables and then push them into an output variables. So all of that is ended up done as a part of your last step. Right. So if I have to just go ahead and in one go execute this corresponding program, we have already had a look at our raw demographic dataset, which looks very difficult for me as a programmer to understand, even though I am in-house. So then the difficulty level is going to be double when you are looking at a regulatory authority reviewing it. So if I have to convert this into a very standardized structure, it would then become very easy for me to view the data. Right, so if I now look at the final data set, I get to see for each and every subject, I get to see the because these variables have definitions already been given. So if I know what these variables, if I know the theory meaning of these different variables, I very straightforward understand saying that, okay, this is the overall clinical trial start date for subject ID 101. So I can then make statements like 101 started the clinical trial on the 2nd of April 2010. The clinical trial ended for subject 101 on the 10th of October 2010. Following this, I can also then look at the corresponding treatment start dates. So again, for this example, clinical trial, there's no follow-up period, there's no preparatory period coming into the picture, and hence our treatment start date and treatment end date is very much pointing out towards the overall clinical start date and the overall clinical end date. Beyond that, again, since I know IC stands for information consent, I know when the information consent sign was formed. Now, this date is obviously going to be prior to the treatment start date, right? Or the overall clinical trial start date as well. Then again, I know when my participant has ended the clinical trial, which is my participation end date. Then I also know that this participant did not die because the death date has not been entered and my death flag is also marked as N, which means that the patient has not died during the course of my clinical trial. Then I have the birth date value for the corresponding patient. I have computed the age on the basis of the birth date and the overall start of the clinical trial. Now it becomes easy for me to figure out the sex and the race. So from the raw data, I could not figure out the sex and the race that easily, but once when I have translated it into the standard or the harmonized structure, it becomes easy for me to understand it. And this is going to save in a lot of effort at the end of the regulatory authority because they are very well aware about the entire understanding of all the variables that get into a specific domain. Subsequently, it's also easy for me to understand my treatment-based variables now. And I do know that this being a multi-center trial, it still has been conducted in the United States of America. All right, so it's a pretty easy way for me to go about doing this activity. The only caveats to it is one, as a SAS programmer who is going to work on preparing the data for a clinical trial submission, I need to understand two very important aspects. The first aspect being I need to have a thorough knowledge of my SGTM guidelines. Only if I do have the thorough knowledge of my SGTM guidelines, I would then be able to figure out which raw variable gets into which corresponding um, submission based variable. So before we could wrap up this particular discussion, let me also go ahead and show you how that mapping aspects takes place. All right. Right. So here you do notice that I have opened up an Excel file and I am going to look at the submission data relating to the demographics, which has the patient characteristics in terms of age, sex, race, ethnicity, death date, clinical trial start date, end date, the treatment definitions, and so on and so forth, right? So I have a two-letter abbreviation. Now, each of my domains within the SGTM are named only with two characters. So the demographics is named as DM. Now, when I select this and I go ahead and say, okay, for my clinical trial, since we're talking about the clinical trial, let me also just show you which is the example that we are looking about. So we are looking at a phase 2B double-blind, multi-center, placebo-controlled parallel group trial of analgesia HCl for the treatment of chronic pain. And hence, given that, it is double-blind, that's the reason why you do have a randomization coming into picture. And randomization date is also available in our source data for our patients, correct? Now, when I get into the variable metadata, here you notice that there are about 23 variables that I need to include in my submission. And all these 23 variables have the variable names being defined. They have the type being defined, the length being defined, and the label being defined. Now you would end up having a question saying that, hey, from where did this information come in? Let me open up another document for you. And that document is the study data tabulation model implementation guidelines. Now, within this implementation guideline, this is something that you can always reference to on the CDISC website. This becomes your Bible for any programmer who has to work on the creation of these domains. Now, here, when I get into section 5 that talks about models for special purpose domains, I have a subsection relating to demographics. When I get into demographics, and there you go, you have a specifications table that details out each and every variable with the core, and any of your variables that have been marked as required, that is something that you would have to bring them in. Whatever has been marked as expected, again, you have to bring them in, but situationally conditional. And then you also do have a list of variables that act as permissible. Now, each variable has a very specific variable name, has a very specific label, and the data type as well is very specific, which means that the data type is either character or numeric. And you would have this exact same definition, which ends up being used in your submission as well. So when you have your list of variables that you need to plug in values for, you then start mapping it towards your input data, right? So you would want to identify which corresponding source table you would want to use for this information. And within that source table, are you looking at one column or are you looking at a combination of multiple columns to be used? Right, so here you do notice that your treatment start dates and your clinical trial start dates and end dates, all of this information is being picked up from the dosing raw table. Right, so this becomes your second very important activity before you can go ahead and start writing the code in order to create your output data set. All right, I'm going to take a pause here. So this is an example relating to how do you go about and prepare your data. And that's the first step in terms of accessing the data. So before we move on to the next piece, which is exploration, since we have reached a logical break, any doubts, any questions still here? In the meantime, I'm also going to go ahead and review the chat window once. Okay, so I do see some questions coming up. What are the certifications we need to do to become a health analyst? So Revati, your question is noted down. That's something that I will pick up towards the end of our discussion today. And duration of the certifications. Okay, that's also something that we're going to talk about. Can you also provide how do the SAS certification data analyst roles? So if I understand this question correct, Dipani, there are multiple different roles in an organization when it comes to analytics. So you can be a part of two very large teams. So you have one team relating to data management, and you have one team which relates to statistical analysis, right? Or your group of people who work on predictive modeling or forecasting and so on and so forth. Now you have two options. You would first want to choose whether you want to get into data management, or you would first want to choose whether you want to get into data analysis and reporting. And this is not something that I am talking about in terms of clinical trials, but I'm making it more generic in nature. And when I say generic in nature, it can be used under the 4 different areas of healthcare and life sciences, which is clinical research analytics, real world evidence, pharmaceutical manufacturing, as well as pharmaceutical commercial analytics. So what we just discussed about, which is relating to preparing your data, that is very specific to the data management team. Now, if you want to get into the statistical analysis team, or if you want to become a hardcore data analyst, you need to have a basic knowledge about data management. And then following that, you would also be interested to pick up the knowledge relating to the next 3 sections of our discussion, right? So certifications again. You've got certifications relating to each of the different areas, and that's again something that we're going to talk about towards our end of our sessions. All right. Mm. Yeah, so I will walk you through the Pani through some of the certification prep guides and where can you find a lot of this information on our SAS websites. So that's something that we are going to do. All right. Okay, what was the point name for step 5 in preparing data sets? Right, so the last point that you are looking at, or the step 5 that you are looking at when you talk about preparing data, is to finally pick up all the processed data and push it inside a submission domain, right? So given that here you're very specifically looking at one particular submission domain, the way it looks like, so you have very specific variables in which very specific information in a very specific format needs to sit in. So your last step ends up looking at utilizing that shell which you created as a part of the first step to take all your variables and just push it into those very well structured variables with the help of assignment statements in a data step. Right, so that's the last step that we had spoken about. Okay, I am more interested into data management, which certification is better for data management. Thank you. All right, so the bunny, that's something that we are going to talk about towards the last 30 minutes of our session where we're going to have a wrap up. I'm going to give you a walkthrough in terms of what are our learning paths that exist out there. And then subsequently, we are also going to talk about what certifications. So I'm going to walk you through the certifications content in the SAS.com website. Those are certain links that I will share with you. And then also the very specific learning paths associated to them. Right, so please hang around till the last 30 minutes of our session where we are going to talk about those aspects. All right, any other questions from anyone? And I hope I have addressed all the questions that have come up in the chat window. And I want to tell thank you to Susanna Zhang for answering some of your questions, and her answer are pretty much on the right track. Yeah, so thanks a lot, Susanna, for that. All right, so I hope all of y'all are back here from the break. And I happen to check on if the recording would be shared with y'all for today's webinar, and the answer is yes. So for those of y'all who have joined in today, the recording of this 4 hours webinar that we're conducting for you is definitely going to be shared along with the deck files. The deck file, the PDF handout is something that I'm going to put into the chat window later today. So request you to please download it from there as in when you see it being plugged in. I will also make a mention when I upload it onto the chat window. So the deck file is something, the PDF copy of the deck file is something that you can feel free to download from the chat window when I upload it there. And the recording would be shared out to you on email with a link and the duration for how long it's going to be made available to you all. Right, so a good time to move on to the next topic, which is the second area in terms of exploring our data. So we are going to start looking at two very important aspects here of our analytical life cycle, which is relating to basic explorations with the help of a visual analytics interface that's primarily used for BI reporting and dashboarding. You can also use visual analytics to get a quick view of your data, and you also do have certain modeling techniques available in there with a visual look and feel in order to give you not very hardcore modeling options, but then a quick, crude approach to looking at or getting insights or understanding dependency information from your data. Right, so to start off with, for visualizing your data, we are going to take up an example relating to the Framingham Heart Study. Now, those of you all who have been associated to the research area of healthcare and life sciences, especially if you do belong to academic institute, then for sure you would have come across this heart study. There's also a link which has been plugged in here. You can definitely have a look at this link to get further information. The data that has been uploaded onto the link is also open source. It's publicly available. So you can definitely utilize that data to go ahead and either uh, use it from a reporting standpoint, or you can also use it as observational information that you would want to then plug in to a predictive modeling technique. Right. So the Framingham Heart study, the main purpose of it was to identify, this had started way back in time, and it had started off in order to identify common factors or characteristics that contribute to a cardiovascular disease. So like most of you all do know, when you talk about any kind of cardiac related problems or conditions or even diseases for that matter, they are very strongly associated to generations. It is considered as a hereditary problem. Over and above that, also different lifestyle conditions that lifestyle options rather that we sign up for as on today could be a factor or a contributing reason to a sudden start of a cardiovascular problem. So the very typical standard lifestyle choices in terms of having a very sedentary work life, or in terms of not having a balanced work and personal life, like in terms of not having enough of exercise or not having enough of physical activity, that could also be one of the causes of an onset of cardiovascular diseases or problems. Then the lifestyle choices of a very regular consumption of alcohol of substances. And when you talk about substances here, it could either be relating to tobacco or any of your hard drugs as well. So those could be, again, lifestyle choices that could possibly contribute towards a cardiovascular problem. Then you also do have certain other factors, like in terms of certain autoimmune conditions. So if you are familiar with a lot of skin problems out there, or if you're looking at lifestyle problems such as cholesterol or blood pressure problems, or if you're also looking at diabetes, then these could also be one of your contributing factors to cardiovascular problems, right? Now, the Framingham Heart Study is looking at studying all of these different factors or characteristics over an extended period of time. And when you talk about an extended period of time, you're not just studying one generation, but you're looking at a multi-generational study. So it started off with the aim or with the idea of studying up to two generations and looking at how these different factors can also influence the lifestyle choices of the subsequent generation within a family or within a specific community or population. And you're also then looking at how those factors can be hereditary in nature, how different conditions that people have suffered from in the past are going to subsequently impact your subsequent generation. Right, so it's considered as a multi-generational study, and the moment you talk about multi-generational studies, it also comes under the very broad area of longitudinal studies in not just healthcare and life sciences, but then you have a similar study design available across different industries out there. One of the best parts is here you're not limiting it to one set of population, but then you're trying to include diverse populations as possible. So again, it brings me to the point that the demographic factors that we spoke about in the clinical trial example that we picked up previously is also going to be the similar thing that you would end up doing when you look at a observational study, right? So a lot of your demographic factors are going to have an important standpoint in defining how a person's health can be factored in or measured onto a particular scale, right? Now, when you look at all of this different data, there would be certain quick calculations that you would want to do in order to visualize the information. So say, for example, when you're looking at age. Now, let's take up a classic example here. When you end up looking at, um, back problems or shoulder problems or any kind of sports injuries, or if you end up looking at the health or the strength of your bone over a particular age timeline, you will not find a lot of prominent difference between an individual or between a human who is 18 years old as compared to a person who is 19 years old. So the bone density in the human body or the fragile measurements of a bone in the human body is going to be pretty much in the same range. If you're not looking at any other health problems, it's going to be in pretty much in the same range of standard values for an 18-year-old and a 19-year-old. So very rarely you do hear about an 18-year-old or a 19-year-old or even individuals lesser than that age having a knee problem, or who have gone in for a kneecap replacement, or who have an ankle injury, or even if you start talking about some of your sports injuries like an ACL tear, which is very common in a lot of racket sports, players, or players relating to football or hockey. An ACL tear is a very common injury, but then you do not find these injuries happening for an 18-year-old or a 19-year-old or individuals lesser than that. But then the moment you start talking about a 30-year-old, or the moment you start talking about a 35-year-old, or even if you're getting more into the senior citizen age categories, then you start hearing about the number of back problems being more prominent or more in prevalence. Then you start talking about kneecap surgeries or replacements happening more frequently, more of ligament fixing that happens at a later age group. And these are some of the reasons why you also do see in different tournaments and sports, you've got age categories, right? So you've got some of the sports you have under 19s, then you have under 21, under 30, under 35, then you've got like an open category, then you have veterans category. And the second most important reason as to why you have these different age bifurcations is because, for obvious reasons, the performance of a player when that player was 18-year-old is going to be very different from the performance of the same player when the person now turns out to be 40 years old. So age as a value, I might not want to consider it as a continuous measurement, and I might not get a lot of analysis if I'm going to leave it like the way it is. So I would then want to convert it into certain groupings. So that's something that you would want to do. Similarly, certain vital signs or certain physical examinations or body measurements have different units of measurements across different geographies. Now, classic thing in India, when we end up measuring weight, we end up measuring it in kilograms. But then the moment you talk about a weight measurement in the United States, you would want to measure it in pounds or lbs as a unit of measurement. And then if I have to compute the body mass index, and on the basis of the body mass index, if I would want to then define a person to be normal, underweight, overweight, or obese, then I would need to have the weights and the heights of my participants on the same unit of measurement. So some of these quick calculations, like the conversion of a unit of measurement or the categorization of ages into different age groups, can very be easily done in visual analytics, and you do not always have to resort to programming in order to do so. Following this, there would be a lot of other variables that you bring into the picture. Like in our Framingham Heart study, the sample of the data that we are going to take, we are going to look at the age at which a CHD was diagnosed for a patient. Then along with that, there would be very important risk factors that you would also want to attach to it. So the weight status, the blood pressure status, the cholesterol status, the smoking status. Smoking also is a very important risk factor that can be associated, or that is rather very commonly associated towards cardiovascular problems, right? So some of these other parameters or some of these other observations for our participants in the study is something that would end up getting recorded. And one of the best aspects of visual analytics is it is very automatically going to bifurcate for you the variables into categorical variables and variables into measure-based variables. So let's take up this example. Let's have a look at how visual analytics can work in order to identify your categorical variables, identify your measure-based variables, how it can very quickly calculate certain variables for you on the go and used in certain quick visualizations. So the moment you talk about a drag and drop based application, everything happens pretty quick. You are not looking at spending time in trying to design things via code. Right, so I am going to use the Explore and Visualize interface of our SAS via platform. So here, if I just show you the platform that I'm using. I am using SAS Visual Analytics in order to create a report. And this SAS Visual Analytics is bundled as a part of the SAS Wire platform. And I am working on the 3.5 version. The more recent version of Wire, which is available out there in the market, is Wire 4. And we do have certain big names in the industry that have started moving onto the Wire 4 platform. Right. So in order to look at a quick analysis of the Framingham Heart Study sample data that I'm taking here, under the Explore and Visualize, I just have to go ahead and click on Start with Data, and then I just need to identify where my data is stored, right? So I can just go ahead and search for the data under the available. And one of the best parts about the Wire platform, here you can see that you have two options. You have data sources that can help you in connecting to external data sources. So you can have data residing in a file location in a folder onto a particular box. You can have data residing in Oracle. You can have data residing in SAP. You can have data residing onto Microsoft SQL Server. You can also have some data flowing in via queues like RabbitMQ or Kafka streams. Now, each of these different sources have different connectors that integrate with the SAS box. So with the help of those integrators, you define the data sources, and all of them end up being listed under data sources. So you can have connections to sum it up. You can have connections to databases. You can have connections to web streams. You can have connections to FTP, which is just, file systems. And you can also have connections to data, which is native to SAS, such as your SAS data sets. Once when you connect to your data, in order to utilize them for your quick analysis, and the reason why here I'm saying quick is because technically, more from an architecture or an infrastructure viewpoint, you are looking at picking up data from your memory rather than the disk. So thereby, your IO in terms of processing and the time that it takes for your inputs and outputs to get generated is much lesser because you're reading it from the memory and your analysis are happening in the memory, your processing is happening in the memory, and you're not looking at the utilization of just one thread, but then you are looking at a multiple parallel processing environment where your data is split across different buckets, and those buckets are pushed onto those corresponding number of threads, and the analysis are done there, and then you see one singular output coming up for you, right? So the starting point would be to have this data loaded onto the memory of your SAS box. And once when you have it on the memory, that is when you as a user can then go ahead and start analyzing it. So here I'm just going to go ahead and pick up the heart data set. And this heart data set, um, I can have three views to it. I can just have a look at the sample data. The sample data is going to show me how the data has been populated across the different columns. Then I can do a quick profiling analysis. And this profiling analysis is going to show me some very interesting statistics for both numerical variables as well as character variables. So I can also get to see the uniqueness of information across these different variables. I can see how much of the information is missing. And then subsequently, I can also get some basic statistics like a mean, median, mode when you talk about numerical variables. And when you talk about certain character variables, such as a BP status or the cholesterol status or the cause of death, these certain variables you can also, you would end up getting just the mode details, because for obvious reasons, you cannot compute a mean or a median for a character-based variable. Following this, you also have certain other statistics like your standard deviations, your minimum, maximum, that also end up giving you a nice analysis in terms of the range of the complete information within that corresponding column. As of now, this details table is not getting populated here, but then this details would actually show you the list of the variables that are available in this corresponding data set. Okay, so please excuse me. Yeah, I just had to click the refresh button. So here you can see the overall description of your variables, so the name of the variable, the label of the variable, the type of the variable. Now the wire data types are slightly different from your traditional SAS understanding of the data types, which would primarily be only character or numeric variables. So here you do see that when you talk about character, you can see character coming up somewhere. But when you talk about numeric variables, then you have double, big int, small int, and so on and so forth. You then have the length-based definitions for each of the variables, and you can also have certain tags. Now, those of you all who are quite familiar with Instagram, Facebook, or X, which is a new name for Twitter, you do see a lot of hashtags there. So hashtags can be used in searching for information as well. So similar to that concept, you also have these tags here where you can use any of this information as tags. And then later on, these tags become easy for you to obtain results with simple search criteria. So in order to get this data attached to a report, all that I need to do is just click on the OK button. And the moment I do that, you notice onto the data pane on the left-hand side, I get all of my data divided into two very important categories, which is all your character variables get defined as category. And here, when you talk about category, you're looking at the understanding of category more from a statistical analysis viewpoint, right? Or a reporting viewpoint. And all your numerical variables will end up getting classified as measures. Following this, you also have certain automatic variables that end up being computed, such as your frequency percentage, because some of the automatic visualizations that you happen to create, such as a histogram or a bar chart, would end up using frequencies. So you can see a frequency also coming up here under the measure. This is an automatic variable calculation. It's not a part of your raw data that you have brought in as a part of this report, right? So you have a frequency measure, you have an aggregated measure such as frequency percentage. These are certain variables that automatically get created in order to have the capability of automatic visualizations onto this setup, right? Now, similar to the basic descriptive analysis that you saw before you could go ahead and click on the OK button, you can get an in-depth measure-based analysis from the report as well. Now, again, in order to perform that exploration, traditionally what you would do, you would write certain programming codes, or you would want to create certain visualization to get that analysis done. But you know what? For your measures, you don't have to do that. In my data pane itself, if I just navigate to view measure details, here itself, I get to see a lot of in-depth statistics for each and every measure variable. So if I'm interested to look at cholesterol statistics, right? So for cholesterol, I get to see what is a minimum value, what is a maximum value. Now 96, okay, fine, but a maximum value of 568, that's definitely pointing out towards some kind of a problem. And the average cholesterol value that I have in my data is about 227. Now I can go down further in this analysis and I can look at a histogram. The histogram can be used in trying to understand whether your data is skewed. Your skewed values can be the positive skewness or negative skewness. And you don't notice that majority of your frequency is lying in the range of say about 200 to a little less than 400. But then you do have certain extreme values that are coming up at 100 and going way above 500 as well. So you can then have a look at your skewness statistic. You can have a look at your kurtosis statistic. You can try and understand if your variable follows a specific distribution or not. And when you talk about distribution-based analysis, you're basically referring to your probability distributions, right? So the skewness statistic is not that large. Although it is positive, there is a slight skewness that you can see in the data, but it is not a very significant skewness as well. Right, so that is some of the analysis that you can do. Similarly, if you would want to have a look at your diastolic blood pressures or your systolic blood pressures, that's again some quick analysis that you have out here. You want to have a look at your height, then that's again something that you can do. Then smoking, this is in terms of how many number of cigarettes. Now, traditionally, again, those of you all who do come from an academic research background, you would want to analyze smoking in terms of the number of pack years. And when you talk about pack years, you're basically calculating number of cigarettes. Each pack contains about 20. That would be the average that you end up looking at. And then you are looking at for how many years. So you end up deriving an average in terms of each participant you're first asking two very important questions. How many number of cigarettes do you smoke per day? And then you're looking at for how long have you been smoking? And once when you get these two answers, you then compute a smoking pack years, which is a much more better variable to use in the analysis as compared to directly going ahead and utilizing a raw variable such as how many number of cigarettes do you smoke per day, right? So this smoking variable that you see in our Framingham Heart Study sample data is just a number of cigarettes that the patient or that corresponding participant has been smoking. So you can see that you've got the numbers ranging from zero, for very obvious reasons, not everyone's going to smoke. So valid. And then you do have the maximum number going up to 60. That's probably pointing out towards a chain smoker. And you also have the average, which is like, around about 9.37. For smokers, not that bad of a number that you're looking at in terms of an average number of cigarettes that you smoke per day. Right. So if you're picking up this variable, again, you do notice that this skewness is much more heavy. So when you were looking at the cholesterol was 0.8, but however, notice there's a skewness here is 1.2224. So this is more of a heavy positive skewness in the smoking variable. And again, there's not a lot of difference between a person who smokes 9 cigarettes per day as compared to 10 cigarettes per day, right? So you might want to categorize this variable. You would want to categorize it in terms of a light smoker, a heavy smoker, a non-smoker, a chain smoker, right? So that would be again defined on the basis of your smoking pack years, or yeah, it's just known as as your smoking pack years. And you do have a lot of standard definitions available out there across different medical research papers that have done meta-analysis in the past to tell you what would be the logical categories that you should be looking at in terms of clubbing that information, right? So you do notice that you've got a small percentage or a small fraction of your sample that do not really smoke any cigarettes at zero value. And then you have the larger chunk of your participants who are looking at smoking cigarettes from probably a number slightly less than 10 and going up to a number slightly less than 40. Beyond that, you have a lesser number of population who are extreme heavy smokers. So this also, if you would want to associate it with the CHD or the age at which the person has been diagnosed to have a chronic heart disease, you would want to club in this information, categorize this information, and then do a cross variable analysis. Right. So that's how you can go ahead and perform in very technical statistical terms known as univariate analysis, not bivariate analysis. Please note this can be used as a starting point for your univariate analysis, and then you can take decisions in terms of, do you want to categorize any of your variables, or do you want to go ahead and use the variable like the way it is in the analysis, right? So we are going to look at two examples in terms of rather one example. I'm just going to take a quick example in terms of how do I go ahead and calculate a variable such as height in feet. Now I know that if I have to convert, the conversion logic from inches to feet would end up being 12 units. So I can just take the height value in the inches and I can divide it by 12, and that would give me the height measurement in feet. So I can just go ahead and search for the height variable. Here I have only height, which is the height being measured in inches. Now, in order to calculate the feet, I just need to go ahead and click on the new data item. And since it's a calculated column, I have an option which is calculated item. I can give it a name. So I'm giving it a name of height. In parentheses, I'm going to go ahead and say feet. And the logic that I want is a division. So the first thing that I can do is from the operators, I can just search for the division symbol. I bring this in and place it. So you do notice everything can be done via drag and drop. If you do understand code, then you can also go ahead and type in the code in the text window. Otherwise, there's no requirement for you need to know code in order to use this tool to do a quick analysis, right? And then from the data items, I will pick up my input variable of height. I would put it into the first parameter, which is my numerator x, and then I would want to divide this value with a 12. Now, if I come to the text, I can just see the code that ends up being generated for this. So you do notice this is a name literal, which means that you're referring to the column that is named as height. So you're doing a height divided by 12. Now, once when this logic is plugged in, you do notice no errors are coming up. So messages are 0. You can go ahead and click on a preview. You get to see for about 50 observations. If your height is got calculated appropriately, you can do a manual calculation to cross check if your values are right. So here we ended up using the logic, which is height in inches divided by 12, right? So for the very first observation, if I have to just do this, 62.5 divided by 12, if I'm going to manually do this, then I get a value of 5.208. Now this value has ended up being rounded to 2 decimal places, so that gives you 5.21. So that's perfect. I do not have any kind of problems in my calculation. This works. So I can go ahead and click on close and click on the OK button. And I have a new variable that ends up being calculated. And how do I know it's a new variable? If you do notice, there's a small calculator as a symbol appearing onto the scale. So this is an indication saying that, again, this is not a part of your input data set. This variable has been calculated as a part of usage for this report. Now, if I just want to look at the distribution of this variable, I do not really have to come to the objects, select a histogram, and then attach the variable to it. Very coolly, I can just pick up this variable, put it onto the canvas, and I get to see the histogram coming up for it. By default, your intervals get defined. It is system defined. If you notice this here on the right-hand side, you have something known as your bin range. But then if you do not want to go ahead with the system defined bin range and you want to manually calculate it, you can go ahead and do it on the basis of the measure values. So you can choose your options according to your convenience. Right, so over and above this, there are certain other visualizations that you can create. You can either create them as an automatic visualization, or you could also go ahead and drag and drop a particular object from the canvas and then create that corresponding object. So this is an example of a comparison of the systolic blood pressure to the diastolic blood pressure. And this helps you in determining whether a person does have a blood pressure problem or not, right? So higher the systolic blood pressure, you kind of do expect that your diastolic blood pressure is also going to be on the relatively higher side. Right now, you can also go ahead and just look at a simple bar chart. And this is what I was saying. The smoking status variable is already being created for us, where you've got participants that have been bifurcated into non-smokers. So the number of cigarettes that they would end up smoking would be 0. You've got your light smokers, which is 1 of 5 cigarettes, moderate smokers, probably 6 to 15, very heavy, greater than 25, and heavy smokers are 16 to 25. And sometimes you can also have missingness of information, so you can get to see that missing information as well. Following this, like I said earlier, for some quick, crude analysis in terms of supervised learning or unsupervised learning, you do have certain objects that you could also use in the visual and analytics interface itself. So there's an example of a clustering algorithm where you are creating clusters on the basis of the weight status, the smoking status, sex, and the cholesterol status. Now, the reason why we are just looking at a quick clustering analysis here is for you to maybe have a targeted treatment approach. So like we made a mention in the clinical trial example, with the example in terms of the dietary patterns being different across the two halves of the India country, then your suggestions in terms of the dietary patterns to be changing is also going to be more tailor-made to that corresponding category of patients, right? So similarly, if I do have patients who are on the heavier side of weight and who have a higher number of cigarettes that they are smoking, and in case if they are females, you would want to suggest in terms of, hey, listen, if you do not want to have maybe your female problems in terms of pregnancy or in terms of menstrual cycles, you would want to watch your weight, you would want to reduce the number of cigarettes. But however, the suggestion when it comes to a male would be slightly more different, because for a male, you're directly looking at, you know what, cholesterol problems or watch out for cardiac problems. So the approach in terms of the suggestions for a problems would be very specifically tailor-made towards the biological sex of female as compared to the biological sex of male. Right, so I'm just going to go ahead and open up a report that I have already created as a quick view. So I'll just come back to the menu. And here I created a very similar report that you see onto, the presentation deck file. It's not a very full-fledged report as such. So here you have, what I've done here is I measured the weight status across the height, but height would not be a correct measurement to use in this case. So again, what I can very simply do is I can just go ahead and change the height to maybe frequency. So I can then look at how many of my participants are overweight, how many of my participants are normal, how many of my participants are underweight and missing. And you do notice a very large percentage or a very large number of my sample is overweight. Now, if I do not want to go ahead and use a status like, a measure like a frequency, I can also use a percentage. So it's very easy for me to change my measurements in one go. I do not have to delete any objects and recreate them. So here I do notice that my sample has about 68%, approximately 68% of my sample is overweight. And a smaller portion, which is just 28 or 27% of them are normal, and underweight are an even more smaller percentage, which is just 3.47. Now, this is a very interesting categorization. So I would want to see how this is impacting, or how is this working with the age at which the CHD was diagnosed, or I can use this in terms of marrying this information to the smoking status, or I can use this in terms of looking at the blood pressure status as well, right? So I can very nicely create simple visualizations that can describe my sample for me. And then following that, I can get into certain quick analysis wherein I can create clusters in order to try and identify if there are certain distinct clusters that are being identified in my sample, right? So here, actually, if you have a look at this, in my analysis, there's not a lot of distinct clusters that's coming up across the combination of variables relating to the weight status, smoking status, the sex, the cholesterol, and the blood pressure. One interesting thing that you will notice here is from your cluster 1, you have majority of your observations from cluster 1. So all the subjects or all the participants who are getting into cluster 1 are having 2 or 3 different weight status. But if you are looking at this entire cluster 1 to have a distinct viewpoint, then you do notice majority of them are having a sex of male, or many of them are your male participants, as compared to a small number. So you have a very small number, you can just select this cluster, and you have a very small number that is getting into the female participants list, right? And for obvious reasons, again, you would see an equal distribution across, your cholesterol status and your blood pressure status as well. So there's not a lot of distinction, clear distinction that you're getting across these 3 different clusters, which brings me to the point that maybe what you might want to do is you might want to utilize certain other variables in your data in order to create those categories if you would want to tailor make a treatment towards each of them. Right now, again, this cluster ID of 3, you do notice here that a major chunk of your participants are overweight. They are non-smokers, they are females, but they still do have a high cholesterol status and a high blood pressure status. Now, this high cholesterol status or the high blood pressure status can directly be attributed towards the weight status of being overweight. In this case here, the significance of the person being a smoker might not come into the picture or might not be that prominent of a factor as compared to the number of observations that have been identified in these clusters to be non-smokers. So such kind of quick analysis is something that you could do with the help of visual analytics, and then you can also go ahead and create a full-fledged dashboard in order to represent the entire sample that you're using in your study. All right, I'm going to pause here before we move on to the next topic, which is, again, we are going to start looking at certain programming-based examples for, again, some of the very common statistical methods that end up being used in research under healthcare and life sciences. So before I move on to this, quick check, any questions? Okay, so there's one question here. Does it auto-generate the text from the drag and drop? Yes, it does. So the text that you see on your visualizations, so let me just take this, cluster example for you, because here you do have a lot of text being shown as compared to some of these visualizations here. So if I just go ahead and from the objects, I can just search for a cluster object, or I can also scroll down to the category of visual statistics. I can just drag a cluster object here, and under the data roles, I would just want to select a couple of them. So I'm going to select the same variables like blood pressure status, cholesterol status, sex, smoking status, and the weight status. And I'll go ahead and say, okay. Now, the moment I bring this in, the number of observations that we use, the number of observations unused, polyline numbers, these numbers that you see here, or whatever text that you see in the screen, all of this is auto-generated. You can also change some of the information here. Along with this, what you also get, if you maximize the object, down you do notice a summary table coming up. So you can get to see summary information in terms of your cluster algorithms, which cluster algorithm was used, what are your centroid statistics, model information, how many iterations have been used, what is the frequency distribution across the categories of each of your different variables, as well as further understanding your parallel coordinates plot, which is this one. Right. So in the previous example that I was showing you, you noticed 3 clusters, but here by default, you have 5 clusters coming up. So if you would want to change this, you can again just get into the options very easily. You can change this to 3 clusters. And when you talk about the text under the object, you can change some of your text. Right. So these are certain things that come up automatically, and you can also enhance the look and feel of it according to your requirement. And since another very important point, since you're looking at the wire box being a multiple threading parallel processing system, for some of your predictive modeling techniques, irrespective of, predictive modeling and unsupervised learning techniques, this would come under unsupervised. You would want to give in a random seed, because this random seed is going to ensure that each time the clustering algorithm runs in the backend, you have the same observations being split across the different threads or the different data nodes rather, right? So this, or data nodes as well, if you have a multiple box system at the backend. So this seed number that you see here is going to ensure that you get the same results across multiple runs. However, if you don't put this seed number, there could be a possibility of your results changing. Okay. All right, sorry, oh, yes, the program code also is auto-generated from the drag and drop. Yeah, that's something that you do not need to type in. You have an option to type the text if you want to, but if you use a drag and drop, it gets automatically calculated for you. Let me just go ahead and open the previous report that I was working on. So here we had height, feet. So if I go ahead and edit this, this was something that we had calculated with the help of the drag and drop feature. If I get into the text, this text gets auto-generated. I can also choose to edit this. There you go. I deleted it. I can also undo it, and I can add further information to this. So say, for example, just as an example, what I'm going to do is just do a height divided by 12 into 1. So it's pretty much the same logic that was written earlier. So this is the only addition thing. So I can also type in the text this way. And anytime when I type in text as well, in the visual, you will notice that taking effect. So it acts both ways. You type in text, you will be able to see the changes coming up in your visual in the drag and drop formula. And if you make any changes to your visual, then that is something that you would be able to see in the text as well, right? So it works hand in hand. All right, perfect. Okay, any other questions coming up when in terms of this quick demonstration relating to visualizing your data? All right, so we are going to move on to the next topic where we are going to talk about the common statistical methods used for research. And the moment you talk about healthcare and life sciences, you are looking at more of a hypothesis testing. And if you do notice a whole chunk of different statistical analysis that has been written down here, starting from your one sample t-test, going right up to repeated measures analysis, then the analysis of covariance going in fact right towards the end. So you have a whole bunch of hypothesis testing that happens when you talk about healthcare and life sciences. And very specifically, you have more of the non-parametric tests of analysis that take place as compared to the parametric tests of analysis. Now, the very common way, or rather the more industry sort approach in order to perform this analysis would be via programming. Very rarely you would find a brand in the industry that uses a point and click application in order to generate these test results. And the reason being that it's any kind of customization that you would want to do for these analysis, so much so as changing a level of significance is very easily done with the help of a code rather than trying to do it with a point and click approach. Second, you do have the same statisticians or the biostatisticians who go ahead and perform this analysis who are very comfortable doing it on R or who are very comfortable doing it on Python. So the two more common languages that are used in the industry for statistical analysis or rather biostatistical analysis would be either R or SAS. You don't have a heavy usage of Python as such. Python is used more for your modeling analysis, which happens to be more prominent in banking, finance, retail, energy, utilities, and so on and so forth. Those other industries. Healthcare and life sciences is still a very traditional industry that needs to have explainable results, and that explainable results, a starting point for it becomes your hypothesis testing. So some of the examples that we are going to take, we're going to take just two examples, and the two examples are going to be a programming example relating to a two sample t-test and a one-way analysis of variance. We are going to look at what procedures are used for this, and you will notice that it's just the very basic analysis can be achieved with just 4 lines of code or 5 lines of code at the max. And for your reference, you do have all the other procedures that have been listed in this that are used across the different statistical analysis. So PROC is a short name for procedure. In SAS, you have only two processing steps. One is a procedure that can draw results and majorly used for analysis. And you have the second step, which is a data step that helps you in creating data sets or manipulating your source data or creating your analytical base tables and so on and so forth. So when you talk about over and above the two sample t-test and the one-way ANOVA example that you and I are going to look at, when you talk about repeated measure analysis and you have procedures such as known as GenMod, then you've got a regression procedure or a GLM procedure known as your generalized linear model procedure that can be used for linear regressions. You've got a logistic procedure that can be used for logistic regression. When you talk about some of the non-parametric analysis or analysis of covariance, which builds over and above the analysis of variance, you can again use your GLM procedure to do so. And some places you can also go ahead and use a very basic statistical procedure such as a univariate analysis. Following that, most of your non-parametric tests such as your Kurzweil, Wallace, Wilcoxon, rank sum tests, these can be achieved with the n power one way test, and the full form of n power one way would stand for non-parametric one way analysis. Following that, your categorical data analysis in terms of hypothesis testing, such as your binomial test, chi-square test, Fisher's test, McNamara's test, Cochrane-Mantel-Hansel test. These are the different tests you can perform with one single procedure known as your frequency procedure. So your procedure block would end up just being 4 or 5 lines of code, but within that 4 or 5 lines of a procedure block, you would have different statements or different options that would enable you to either perform a chi-square or perform a Fisher's or any of the other tests that have been listed down here. Over and above this, when you talk about survival analysis, which is heavily used in the industry, in healthcare and life sciences, especially under research, you have a very specific procedure known as the proportional hazards regression model, right? So that's something that you can do for a Cox proportional hazards model. In fact, this PH reg procedure has certain statement options. It's quite a complicated procedure, but it does have certain statement options that you could possibly use if you are also looking at non-proportional hazards model, right? So it's not limiting you just by for working with proportional just because the name says so. Don't get confused by that. But then that's one procedure that very well works for even non-proportional hazards model. So we are going to first start off with the example relating to the two sample t-test. Like, here, the moment you talk about two sample, you're basically doing a two group comparison. And we are just talking about a simple test, a drug, which is ABC123 to have any effect on FEV1. Now, FEV, like you all know, stands for the forced expiratory volume. So you're looking at your samples would end up being people who have respiratory problems. It could either be asthma or it could just be a simple breathing issue. And it doesn't have to be a very focused asthma related sample, right? So you're just looking at, in case of a person who is suffering from any kind of a respiratory problems, if they happen to consume the ABC123 test drug, is there going to be any difference between the forced expiratory volume prior to administration as compared to after the administration, right? So in this clinical trial, very specifically, you are looking at chronic asthma. But like I said, when you talk about treatments for respiratory issues, they do not always have to point out towards asthma as of today. The sample that you and I are looking at is for ABC123 relating to a long-term treatment of patients with chronic asthma. The treatment has been defined as a double-blind randomized clinical trial where you have the oral doses of ABC123 versus a placebo, which is a treatment of zero effect, being assigned to the patients or administered to the patients for about a 6 weeks interval. The primary measurement is the resting forced expiratory volume before the 6 weeks and at the end weeks of the 6 weeks of the treatment period. So you would want to compare the before and the after treatment in terms of the changes of the forced expiratory volume. Now, in order to look at this analysis, you end up doing a very simple. Now notice this, you just have 4 statements, but this is the most basic of the analysis that you would do. So you just end up writing the t-test procedure, proc t-test, you point it out towards the data. And since you're doing a 2 sample t-test, your 2 samples become one that has been administered the ABC123 and the second one which is being administered the placebo, right? So that becomes your 2 groups under the class statement. You would want to do a comparison of the change variable. Change would be the difference between the FEV1 prior to the treatment and the FEV1 at the end of 6 weeks. So the difference between those two values is what gets captured as a part of this change variable, right? This format statement is a very generic statement that has been applied. So this is something that you can keep aside. But then if this simple analysis is something that you would want to run, let me just pick up the code for it. We are looking at a 2 sample test here. Yeah. And let me come back to SAS on demand for academics. All right, so in the interest of time, I have already written the code. So please don't mind if I'm just going to directly go ahead and execute them. So there's something that I'm going to skip. I'm directly going to, let me just comment this out. Okay, so directly going to come to the means procedure. Means procedure is more of a summary procedure that you could use in order to just look at your values for different variables, numerical variables, by grouping them by a specific class variable. Following this, we have applied the same t-test procedure you saw on the presentation deck file, where we are going to perform the 2 sample analysis with the help of the treatment group class variable. And we are going to analyze the change variable, and the change variable is a difference, which is FEV6 minus the FEV0. So if I go ahead and execute this code, first let's have a look at the means procedure output. So here itself in the means procedure, you can see that you get 2 different analysis, one for ABC 123 and one for placebo. Across the 2 groups, we have about 11 participants and 10 participants respectively. If I have to look at the mean of FEV0, it is 2.48 in ABC 123. FEV6, you see 2.98. The change is about 0.50. So yeah, the forced expiratory volume is definitely gone up by 0.50 points. If you're looking at the placebo group, FEV0, which is prior to the start of the treatment, is 2.49, and the forced expiratory volume has gone up to 2.77. And the change between the two is just 0.28. So if I have to manually look at the analysis between these 2 outputs, then I do notice that for the treatment group of ABC 123, the change has been higher as compared to the change for the placebo group. Now that's the analysis that I am drawing. I would obviously want to validate it with the help of an official hypothesis testing at the default significance level of 0.05, which is 5%. So on the basis of the t-test, I can then start looking at the comparison. Now, before I could go ahead and actually look at the difference in terms of associating it to a significance, the first thing that I want to do in a 2 sample t-test is check for the equality of the variance, right? So when I talk about the equality of variance, you have to first figure out if the standard deviations between the 2 groups, are they the same or not? And here you do notice that for the change variable, you can see the standard deviation to be 0.51 in ABC 123, and the change variable has a standard deviation of 0.50. So there's not a lot of difference in terms of the variances. So the equality of variances are also going to point out towards the same value. And then I conclude that it is equal variance. And on the basis of the equal variance, I go ahead and perform the hypothesis testing on the pooled method. And here I do see that the probability of T is 0.3425. So again, this is an indication that this is not pointing out to a major difference between the 2 groups, right? So since your probability of T here is 0.3425, it is greater than 0.05. You can also look at the distribution. Notice that your distributions are pretty similar. Your boxes are pretty similar. You can also conclude the same thing with the help of the the confidence intervals of the mean. So the mean of 0.5027 is very much a part of the placebo group confidence levels of the mean, and the 0.2840 is also very much a part of the confidence levels of the mean relating to ABC 123. So there are multiple methods via which you can conclude And that manual comparison, which I was doing, saying that, okay, the 0.50 and 0.28 might not be that significant of a difference, or it might be that significant of a difference, can be tested out with the help of your t-test procedure. Now, this analysis is being done at a default significance level of 0.05, but that might not necessarily be the case when you talk about healthcare and life sciences, right? That's too strict, and you might want to increase your error significance level, make it probably 20% or 30%, and then do a comparison. All right, so multiple ways that you can perform an analysis with the help of this code, and then over and above this, if you would want to change certain options or you would want to change the approach, you have different options, different statements that you can use as a part of the t-test procedure. Now, in a similar lines, let's also take up the one-way analysis of variance. This is slightly more in-depth as compared to just a simple hypothesis testing. And here we are looking at an example of an HAMA, that's a questionnaire, a standard questionnaire, which is used in order to assess the general anxiety disorder of our subjects. And here we are looking at a serotonin uptake inhibiting agent, SNX 95, which is being studied in subjects with general anxiety disorder. Now, our subjects have either moderate or greater severity of the general anxiety disorder. And again, they have been randomized into either one of the three treatment groups. And it's a double-blind study. Double-blind meaning neither my participants nor the doctors know which treatment group has been assigned to which patient. Now, for obvious reasons, for anxiety related condition, you might or not might, you will want to have a blinded study, right? If not for a double blind, at least a single blind study is something you will need to have, because the way a subject thinks about whether they are being treated or not, that could also be a very important point in uh, you know, controlling the anxiety of a patient. So there are three treatment groups here: 25 mg of SNX 95, 100 mg, and placebo. So these three drugs have been given individually to different patients independently for about a period of 10 weeks. Now, when you look at the one-way ANOVA, here you are focusing more on using the GLM model. You can also achieve the same thing with the help of an ANOVA procedure. That's another option that you have. You're requesting for a box plot. Uh, your dose group, again, your treatment groups again become a part of your class statement. And the model statement is what you would end up using in order to understand your dependent variable and your independent variable, right? So the independent variable here is the dose group, and the HAMA is nothing but the scores from the questionnaires relating to the general anxiety disorder. So that becomes a dependent variable. Then you're requesting for the means across three different groups. So an individual group-wise comparison with a homogeneity of variance test again, and you want the Dunnett's test to be done by considering the placebo as your reference interval. And then you are also looking at a contrast statement to compare active versus placebo. And here you're looking at different age groups at different intervals, right? So the GLM procedure is again going to give you a pretty similar output. First, since it's just a one-way ANOVA, you would end up getting your dose group differences. You've got three dose groups, your degrees of freedoms automatically would become 2. And here, with the help of either your type 1 sum of squares or your type 2 sum of squares, or even your overall model, you do notice that your probability value is less than the default significance value of 0.05. And having said that, when the value is less than 0.05, you end up concluding that yes, there is a difference between the different dose groups and the HMA scores for those different dose groups. But then second thing you would be interested to understand is which dose groups are showing you that significant difference, right? So you can then, before you move on to the next part, you would want to check for the homogeneity of variance across dose groups. Now here also you do notice there is equal variances across the dose groups, and then you perform a means analysis between your different dose groups. So this is placebo versus low, placebo versus high. High would point out to 100 mg, low would point out to 25 mg. So you can then utilize your difference between the means. You can have a look at the confidence limits, and wherever you have the asterisks being mentioned, those are the points or those are the comparisons that are significant at the 0.05 level. So for obvious reasons, when you compare the low intake of 25 mg versus no intake at all, that is going to be a significant improvement in the HMA scores or in not feeling the anxiety. If you're going to compare high to the placebo, again, it is going to make a difference. However, if you compare the low to the high, notice this: if you're going to give a patient of those 52 subjects who have been given 100 mg and 25 mg, you do not see a lot of significant difference coming up here. All right. So this is very interesting. So your low to the high comparison, 100 mg versus 25 mg, is not giving you a significant comparison. However, 25 mg versus 0 mg and 100 mg versus 0 mg is indicating to a significant change in the HMA scores. Clear with this? Now, before I move on to the Cox proportional hazards model, here we are going to pick up an example either with the help of a little bit with the help of the programming. And I'm also going to show you another very interesting approach. How can you do survival analysis with a point and click option on SAS Studio? So before I move on to that, let me pause to check if any questions that we are looking at here from anybody. All right. Please feel free to drop in questions if you all have any. So I'm now going to move on to the next part, which is a Cox proportional hazards model. Here, I'm not looking at non-proportional hazards, but then we limit the discussion only to proportional hazards. And we are taking up an example relating to the recidivism study, which is a tendency of a convicted criminal to reoffend. So this is an observational study. It's not a clinical trial per se, or an experimental study rather. So in this example, this is just a sample of the data that you see on the right-hand side. Now our sample has about 432 male inmates. Yes, sorry. So that's correct. SK, I'm sorry, I do not know your full name or the name that I should be addressing you with, but for each and every hypothesis testing, you have to check for normality. I mean, that goes without saying. So that's a compulsory check which you need to do before you proceed. So there are two checks rather, a normality test, and then you would also have to check for the homogeneity of variance. Typically, if you have a homogeneity of variance, that is when you go ahead and apply a specific method of the analysis, right? So both the assumptions have to be fulfilled before you go ahead and look at the hypothesis testing. And hence you do have that entire bifurcation between parametric tests of analysis and non-parametric tests of analysis. All right, so coming back to this recidivism study, our sample has about 432 male inmates who were released from the Maryland State Prisons in the early 1970s. Now, this is a published observational study. You can find it in the papers by Rossi and others, published in 1980. These inmates who were released in 1970s were followed for 1 year, and it was checked whether they are going to reoffend and again get arrested. So here you're just looking at only the first arrest. In fact, we are just looking at only the first arrest. So the time after coming out of the prison to being arrested once again. Now the week variable contains a week of the first arrest after release, right? Now say for example, if you are looking at inmate number 8, inmate number 8 was arrested for the first time after release at week number 19. So it took 19 weeks for subject 8 to again convict or to be convicted as a criminal. Now the variable arrest has a value of 1 for those who were arrested So like you can see here, subject 8 had convicted and hence got arrested again in that 1 year follow-up period, and it has a value for 0 those who were not arrested. So if you see this, subject 1 ends up having a value of 0 because a person did not get arrested again. And for those subjects or those inmates who did not reoffend and who did not get arrested again, we have right censored our data to have the weak value to be about 52. Okay, which is the end of that entire 1 year follow-up period. So the overall observation is that only 26% of the men were arrested out of this 432 sample inmates. And the data, like I said, is right censored. And when you talk about right censoring, you're basically saying that if the person was not arrested, which is carrying a value of 0, then the week or the time is going to point out towards 52. Okay, just give me a second, please. We have a battery issue here. All right. Yeah. Sorry for that. Okay. So the next thing that we would want to do is we would want to try and understand our covariates in the data. So there are multiple different input variables that we can look at. So you have the financial aid, whether they do have access to financial aid or not in that 1 year when they were released. Then you would want to look at the age at the time of release. You would want to look at the race again. Certain demographic factors, just race, would be very important in trying to understand the cultural habits. Then you've got the work experience. You have the marital status, whether they were released on parole or not, and the number of prior convictions as well. So if you have a repeat offender, someone like a serial killer who has continued doing the same thing over and over again, irrespective of receiving corrections or not. So that's again something that you would want to check upon. Right. So when you are looking at all of these different variables, you can just go ahead and utilize a live test procedure in order to check for whether certain variables are going to influence the outcome or not. So here you are studying the arresting of your patients, arresting of your inmates, first arresting again after release. When did it occur? And you're censoring it to 0. 0 means they have not been arrested again. And you're trying to stratify this according to the work experience and the parole, whether they were released on parole or not. And you're adjusting this analysis according to the Tucky analysis. So a simple, again, 3 lines code can be written down in order to figure out whether any of your stratas that you have created on the basis of the 2 variables, which is having a work experience before incarceration and whether you were released on parole or not, whether that's going to have any kind of an impact in terms of whether the person gets rearrested. So here, if you end up looking at the work experience, it can carry values like 0 or 1. 0 is pointing out towards 0 work experience before incarceration, and 1 is pointing out towards full-time work experience before incarceration. And parole points out towards 0 if the person was not released on parole. Others released on parole would end up having a value of 1. Now, across a combination of these 2 variables, you can look at how many inmates were there, how many of them failed meaning they got rearrested, and how many of them did not get rearrested. So this is more of a descriptive analysis. And following this, you can also perform a comparison between the corresponding stratum. So you can then have a look at which stratum is, sorry. Yeah, so then you are ending up looking at which stratum is showing you a significant difference and which stratum is not showing you a significant difference. So here you do notice that there is, again, you would want to perform an equality test over the stratas, and only after this you then directly go ahead and look at the stratum-wise differences. Similarly, age as well again acts as one of the variables that you would want to take into consideration in a categorical fashion rather than just directly looking at a continuous numerical variable. So again, you can just directly define your ages in groups. You do not have to use a data step separately. It can be a part of the life test procedure itself. So here you're creating age groups like less than 21. Then you have 21 to 24, then you have 24 to 28, and then you've got greater than 28 categories. So this strata, age, and in parenthesis 21, 24, and 28 is going to end up creating 4 specific categories of age for you. Now those 4 categories of age end up becoming 4 different strata. Across these 4 different strata, you're again looking at how many of them, how many of the inmates belong to that corresponding age, and across those number of inmates, how many of them failed, which means how many of them got arrested and how many of them did not get arrested again. And then you do a comparison between the different strata, right? But prior to that, again, you would want to check for your equality over strata and then decide. Now, notice this. I'm just going to stress a little bit here. There's, for obvious reasons, you do not have an equality over the strata. So if you just look at stratas 3 and 4, then all right, fine. Here you have person censored as 81% and person censored here as 84%. And if you also look at the number of inmates that failed, which means who got arrested, here there is a similarity. There's not a lot of difference between 16 and 17. However, when you look at stratas 1 and 2, which is less than 21 years of age, and the second strata, which is 21 to 24 years of age, you do notice a very significant difference. So you have more number of inmates who got rearrested after release, 52 of them, which is a good 41% of 127, and round about 29 of them from the second age category. So for obvious reasons, you do notice that yes, there is a significant difference between these two stratas, and hence your test of equality over the strata has failed. In case if this test of equality over the strata has not failed, then you would want to consider them as non-proportional hazards, and there would be certain tweaks that sorry, let me correct myself. In case if the strata, the equality over the strata is failed, like in this case, then you will want to consider them as non-proportional hazards. And when you consider them as non-proportional hazards, there would you would have to do certain adjustments in your PH reg model in order to adapt to those non-proportional hazards. So one of the examples of that would to try and marry age to the time, because it is all relating to how people respond during a particular situation, and the way an 18-year-old is going to respond to a particular situation as compared to a 40-year-old is, for obvious reasons, going to be different. So that comes under a whole category of survival analysis known as your time dependent covariates, and associating your time dependent covariates to the non-proportional hazards. But in this case, I am not going to have a look at the non-proportional hazards. I'm just going to look at proportional hazards. And what we are going to do is I'm going to come back to SAS Studio. I'm going to get into the task and utilities here. And under survival analysis, you have the proportional hazards regression. So rather than writing a code for it, I can just go ahead and open up the proportional hazards regression. I can point it out towards an input data set. Okay, this is on. Point it out to an input data set. This is where the data that this is the data that I'm using, saying that it is the recidivism study from the HA library. The censoring that we have in our data is right censoring. So that's selected correct. Our survival time is a number of weeks that the person did not get rearrested, right? So I'll go ahead and select week. For the status variable, I have arresting as my status variable, and my censoring value is 0, which means that the person did not get arrested, correct? Now, the code still does not get generated because the moment you talk about a proportional hazards regression, you need to have certain independent variables coming into your model statement. Right, so for the explanatory variables, I'll go ahead and click on the plus symbol, and I am going to take into consideration age, the financial aid, age, race, prior work experience, whether the person is married or not, and whether the person was released on parole or not. So I'm just taking the first 6 variables and I'm going to go ahead and say okay. And you do notice that the PHREG procedure ends up getting used. You have the class statement, which is defining each of these different variables because they are all categorical in our analysis, like the way we saw earlier in the data description. These are all coded as 0 and 1 or 2 and 3, and they're all pointing out towards it being a categorical variable. So we should not be treating it like a numerical variable. So since we have added it into the explanatory variables, which is categorical variables, you do notice a class statement coming up here. And our model statement, you have the time as week, arrest is our status, and here we are going ahead and pointing it out towards the censored value of 0, which is pointing out towards not being arrested. And we're looking at if any of these input variables are impacting the arresting, the rearresting of our inmates after release. So I do not really have to plug in anything else. You do have parameterization and treatment of missing values, different model options, overall model options. So there are different things that you can do, but I'm just going to go ahead and execute the simple logic that we have built in. And let's quickly analyze. So now getting into much of the detail here, you can have a look at the design variables. And more importantly, we are going to come down to, the likelihood effects in terms of the type 3 analysis. So here, for each and every effect that we have, we are going to check which of them have a significance. And at the 0.05 level, you do notice that none of them are pointing out towards having a significant impact on the way our inmates are behaving, right? So financial aid was slightly close. It had a value of 0.06, but the rest of them do not really have an impact, right? So that's how you can go ahead and perform quick survival analysis. Now, for obvious reasons, you would not want to use a proportional hazards here because the test of equality over the strata that we were looking at for both the options in terms of the work experience and the parole release, as well as when we were looking at the next one relating to age, both of them have failed the analysis. So you would want to make adjustments for non-proportional hazards in this case. That would give you a much more clearer analysis as compared to the effects that we are seeing right now from a proportional hazards regression approach. Sorry, hoping that all of you all are back here. So we've got just 2 more demonstrations to go relating to the predictive modeling techniques, and that's something that we will try to wrap up in the next 20 minutes. And following that, I'll go ahead and also start discussing the certifications, discussing the different learning paths, and in case if any of y'all do have any doubts or questions, additionally, we can definitely take them up as well. Sorry, I had missed out a question in the chat window. I had plugged in a reply during the break. The question was, if the Levene's test failed, which test do we have to apply? So you would end up using a Levene's test for the homogeneity of variance. And in case, if that ends up failing, what happens is that it's kind of hinting towards a non-parametric analysis, which is needed then. So you have a whole bunch of non-parametric analysis, tests of analysis that you can use. So you've got Kurzweil-Wallace, you've got Wilcoxon sign rank. See, you would have to, pick and choose which one would best suit your corresponding requirement and then go ahead and perform a non-parametric test of analysis, right? So that's something that you would have to do, even if, even when you check all your normality-based assumptions for both ANOVA as well as hypothesis testing, right? All right, so moving on to the next part, the example that we're first going to take, like I said earlier, we are going to be looking at a decision tree Okay, I'll just take up some of the new questions that are coming in later after we complete the predictive modeling path. Yeah. So coming to the predictive modeling, we are going to look at certain use cases regarding decision trees. And in order to work with the decision trees, the example that we are taking up here is predicting the low birth weight. Now, low birth weight, according to the WHO, has been defined as weight at birth to be less than 2,500 grams or otherwise 5.5 lb. Right now, the low birth weight is very important because it's a very valuable public health indicator, and low birth weight is also used as one of the very important factors when you talk about the overall health of a community or of a region or of a country for that matter. Right, it is an indicator of maternal health, it is an indicator of nutrition, it is indicator of the healthcare delivery system in the country, and it is also a very important indicator of poverty. Right, so it is very important to always address the low birth weight, and this is one of the very important reasons why you have the entire health administration, especially when you talk about India, health administration in India is a state level aspect rather than it being a national set of guidelines. I mean, you do have the national body, the Ministry of Health. However, the rules and regulations are more fine tuned towards each and every state requirement. So it becomes a state regulation aspect. Now, when you talk about low birth weight, this is something which is very prominent in more of the underdeveloped sections of the society or the least developed sections of the society, such as rural areas, villages, uh, not your urban places as such, more of the tier 2 or the tier 3 cities, according to the definition of macroeconomic factors. Right. So the lack of public health facilities, a lack of proper nutrition in terms of easily accessible rations of food, or the lack of amenities of clothing and shelter could lead to a lot of reasons in terms of not having a very healthy pregnancy for females, for the biological sex of females, not having a very healthy pregnancy, and it can impact a lot of other subsequent factors. So here, when you are looking at dividing your factors into macroeconomic and microeconomic factors and indicators of poverty, healthcare delivery, that becomes a part of the development factors relating to macroeconomic indicators. But in the moment you talk about maternal health and nutrition factors, these become more microeconomic factors in nature that are then also subsequently going to impact your macroeconomic factors. Right, so it becomes very important if you can try and predict this information beforehand rather than waiting for things to get worse. So the sample of the data that we're going to look at, again, it's a North Carolina based study, and you're looking at the North Carolina births of 2000 and 2001. And it includes about 120,000 births each year. So we are going to derive a sample out of it. So our sample is more of an oversample. It has 50% of records that have a low birth weight issue, and then you have 50% of the births which are a normal birth. Right, so your target variable is LBWT, where LBWT stands for low birth weight. It's 1, which means that the baby that has been born is not satisfying this condition that has been defined by the WHO. And then a value of 0 means that it is satisfying this condition that has been defined by WHO. Now, prior to oversampling, the percentage, in reality, the percentage of the low birth weight is about 7.2%. So again, predicting low birth weight is an example of an observational study, which is used very heavily in government research. Now, the data description, there are again multiple different factors. And notice this, you are not just looking at the medical history, you're not just looking at the prenatal status, you're not looking at the reproductive history or the previous pregnancy history or the live birth history relating to the female biological sex in consideration. But over and above that, you're also looking at the demographic factors relating to ethnicity, age, education, and marital status. Point that I want to bring your attention to is the moment you talk about healthcare and life sciences, You are not just limited to what happens inside the human body, but you're also interested to what happens around the human body, right? So your social, your behavioral, your demographic factors play a very important role in determining your health. So there are multiple different techniques again that you can possibly use here. Now, if you're looking at a modeling technique relating to healthcare and life sciences, you would always want to have a model that you can explain, right? So regression methods are very easy to explain because that's a more important point that you would want to highlight, as in what are the causes of low birth weight, right? So that's something that you might not end up getting from a neural network or from complicated decision trees such as your gradient boosting algorithms or forest trees and so on and so forth, right? But nonetheless, you have a whole bunch of analysis techniques available to you, and you can then decide, do you want to go ahead with a programming approach, a drag and drop approach? If you have moved to the new box, then do you use SAS Wire? Do you use SAS 9? So you have point and click options, you have programming options across all the different versions of the platforms that we have at our end. Right? So we are going to try Model Studio on SAS Wire. And before you go ahead and decide which analysis technique you would want to use, there are a lot of questions that you would want to first ask about your data, right? So the starting point would be, what is the size and the nature of your data? Now, next, you would want to try and ask, what are you trying to achieve? Are you just trying to achieve a prediction, or are you also going to try and achieve a causation along with the prediction? And again, I'm stressing, when you talk about epidemiological studies or you talk about observational studies, especially when it comes to healthcare and life sciences or public health in general, predictions is not your only answer. Causes are also going to be a very important point, right? You would always want to find the root cause of the problem and not just look at whether a problem is going to occur or not, correct? So when you end up defining your problem to be both a root cause analysis along with a prediction, and thereby take decisions in terms of maybe budgeting or redoing healthcare delivery systems in a particular area, you would want to use explainable models such as a regression method or a simple decision tree. You would not want to complicate your statistical model by using more of the fancier models that you have out there. Third, how accurate does your model need to be? And again, when you talk about healthcare and life sciences, there are very specific therapeutic areas where accuracy is going to play a huge role, right? So when you talk about computer vision or image analysis in terms of MRIs or your brain scans, or if you're going to look at ECG analysis, or if you're going to look at, say, tumor identifications, tumor detections, there your accuracy levels would be very high. But in case if you are looking at certain more of the generic causes or generic reasons, uh, you know, since your cultural and ethnic factors are going to play a huge role, accuracy might not always be something that you would want to get after. Very important, how much time do you have to train your model? Now, this is a very important point that would come into picture, especially when you had COVID, right? So now when COVID hit us, and if decisions had to be taken, or if people had to figure out what was a reproductive rate or what was a prevalence or incidence as well. So prevalence basically means that for how long it's going to last in a particular human body. So if you're going to try and figure out these different aspects, you do not have a lot of time in order to utilize a very complicated model. So you don't have a lot of time to train your data, but then you would want some really quick understandings. So that also brings me to the next point, saying that certain analysis or certain questions can directly be answered with simple help of descriptive analysis or hypothesis testing. You do not really have to jump into predictive modeling every now and then, right? And last but not the least, because here you are talking about causes as well, you also need to try and figure out if your model needs to be interpretable or understandable in nature. So a neural network model is not really understandable. But if you still want to use a neural network model, hey, go ahead, be my guest. But attach a regression later on or attach a decision tree later on. Force that decision tree or force that regression to use all the variables that have been identified or all the parameters that have been identified by a neural network to just explain it to get into the root cause analysis, right? So these are some of the questions that you would want to ask, especially if you are looking at healthcare or life sciences based analysis, right? So let's have a look at how does this work. So I already again have a flow ready. It is the NC state AR. So I'm just opening this up. And if you just have a look at the data, it's very easy for you to plug in which variable is a target variable. That would be very important for you to specify. And here, if I come to the role, or if I just go ahead and search for the low birth weight variable, you do notice that it is a binary variable and the role has been marked as target. So here, I can go ahead and also specify the corresponding target event level that needs to be used in the prediction logic, or I can just go ahead with whatever has been plugged in as a default value. So there are a lot of different variables that are acting as my input variables. Some of them are interval variables. Some of them are nominal variables, binary variables, and so on and so forth. So if you see here, you do have certain conditions like in terms of diabetes or cardiac diseases or anemia in the mother or amniocentesis, the presence of amniocentesis or the volume of the amniocentesis or the presence of lung diseases, so acute or chronic lung diseases. So these are certain input variables that have been used. These are all medical based variables. Then you also have the drinking habits in terms of alcohol consumption. Again, certain input medical variables. Then you've also got a lot of different demographic variables. So you also have the father details coming in, like the age of the father, the education of the father, the ethnicity of the parents, very important again. Then you do have a certain other medical conditions such as your genital herpes, oligohypertension, hypertension during pregnancy, what is the outcome of the last delivery. Again, a whole bunch of um, demographic variables such as age of the mother, marital status, education of the mother. These are a lot of variables that are going to also have an impact on trying to determine the low birth weight of a future delivery or the next delivery of a patient, right? So the input data is a combination of two very important things: medical conditions or medical input variables. So like I said, you're not just bothered about what happens inside the human body, but you're also bothered about what happens outside the human body. And there's a very important point that I want to bring in here again. uh, this is a new thing that you have been seeing over the internet these days, which talks about marriages within families or within extended families and how that is impacting the birth of babies, right? Or birth of children. So that's a very interesting study. And those studies also are a cause or are a result of such kind of research academic papers. Right, so here with the help of these medical variables as well as with the help of the demographic variables, we want to try and determine the causes of the low birth weight for a new birth. And you can go ahead and perform simple data explorations. Notice this is all done in a pipeline or in a flow mechanism. So if I happen to look at the data exploration, then I can choose and see what kind of data do I want to work with. Do I just want to work with my training data? Do I want to work with my test data? How do I want to check for the importance of my variables? What should be the selection technique? How many variables would I want to select in the analysis? How do I want to go about working with the interval variables and so on and so forth? So if I just go ahead and right click, since I have already executed them, if I just go ahead and click on the results, I can just get a view of the most important variables in my data. So here you do notice that the most relative input, the highest input, which is associated to the condition of low birth weight in my oversample, is the hypertension during pregnancy, right? So that comes up as the most significant variable, and all the other variables are the relatively important variables when compared to the first most important variables. So you have the presence of eclampsia, prenatal conditions, preterm conditions, uterine results, cervix results, alcohol, education, years last live birth. So a lot of different variables. So for very obvious reasons, your medical factors would turn out to be significant first, and then you would have a demographic factor coming into significance. Now, even when you think about it, your demographic factors are the reasons why you do have certain lifestyle conditions. Those lifestyle conditions are going to show you bad medical test results, and those bad medical test results are subsequently going to impact an outcome, right? So it is not surprising that the important inputs also end up listing down all your medical conditions first as more significant input variables, and then you have the demographic variables that are coming up. So here you notice the father age, the father's education, the mother's age, the mother's education, the presence of alcohol consumption, whether the person, the mother, is a smoker or not. These are some of your demographic variables or your lifestyle choices. And these are going to impact your medical results. If they are going to impact your medical results, for obvious reasons, your medical results are going to turn out to be the most important factors. Now, having said this, if you would be looking at a regression analysis, you would end up identifying some of your demographic factors to be acting as confounder variables, because um, the lack of education for a particular person would not just impact the health condition of the person, but would also at times impact the lifestyle choices that a person would end up taking. Now, this might not be true for a very metropolitan or an urban setup, but it can definitely be true for a rural setup. Let's say you would have to play around with the data in the similar manner. And before you directly go ahead and jump into a modeling technique, you would have to perform your bivariate analysis, see how each of these relevant inputs are working with the target variable. And like we said, you would want to try and also associate your demographic factors to some of your medical results. So you can associate them, you can try and see if they're turning out to be significant. Then you can choose to bring them in as a combined factor. You can choose to ignore one of the two, or you can also choose to bring in those variables as confounding factors or stratification variables in the entire analysis. Right, so there are multiple ways in which you would want to use your data exploration results, and you get a whole bunch of them. So this was just one of them that we looked at. Then you have your class variable summaries. You've got the interval summaries as well, cardinalities. Then here you get to see the skewness, kurtosis, the deviation from normality. So you do have here one, which is a drink number, which could possibly be an identification of the level of alcohol consumption. So it has a very high skewness of 31.959. So you would again want to choose whether you should be tweaking that or whether you should be ignoring it or rejecting it from the analysis in question. So now that again goes hand in hand with your relevant inputs. So here I just see drinker coming up to be significant. I don't see drinking number. Drinking number is also turning out to be a significant or has a relative importance statistic. But the relative importance to the first important variable is a very small number. So again, you can decide if you would want to tweak this variable, convert it into a standardized scale, and then utilize it, reduce the skewness, reduce the kurtosis in the analysis, and then move ahead with it, right? So that's how you go about with just using, simple drag and drop nodes. I'm just going to show you one simple decision tree. So in order to create the decision tree, you can just right click on your data node. You have add child node. And since it's a supervised learning mechanism under supervised learning, I have a decision tree option here. And this decision tree, there are multiple different options that you plug in. So some of your basic options would be how you would want to grow your tree, which is in terms of splitting. So you have different criterions that you can plug in: shade, chi-square entropy. The default is your information gain ratio, if I'm not wrong. Then for your interval target, you have shade, FDIS, and variance. Following this, you have the number of branches that you can specify. Default is 2, maximum depth. Then you have, yeah, these are all your default options. Then what is the minimum leaf size? Minimum leaf size is 5. This follows the same chi-square rule of having at least 5 observations per category. Then missing values, how do you want to go ahead and treat missing values in the analysis? Number of interval bins for an interval variable, what technique should be using, a quantile or a bucket technique? Do you use an input just once, or can you use it multiple times? You would always want to, use it once. What kind of pruning options? You can also perform auto tuning in case if you're completely clueless. And the reason why I'm saying completely clueless is because sometimes auto tuning might take a lot of time in order to generate the result for you. So you would want to use the auto tuning facility only if you are completely clueless about the different options that you should be using in order to build your decision tree, right? So that option is also available for you. And then since it's a simple, understandable decision tree, you do not really have to plug in your model interpretability. These come into place, your global interpretability, local interpretability, and the PD or the ICE options. These come into picture when you use certain other algorithms, such as your forest or your gradient boosting, where the inputs or the model per se is very difficult for you to understand. And let's just have a look at of a simple decision tree. So if I just go ahead and right click on this, since again the run has got completed, I can just get into the results. And here, okay, let it just pop up. Yeah, so you do get to see the tree. You would have to expand and then zoom in. That's something that you can do. Then you can, or like, let me just go ahead and zoom in here. Yeah, so if I have to scroll right towards the starting, then these are the total number of records that we have. And here you are trying to predict for one, which is causes for low birth weight. And the very first, which is coming out to be significance, is the presence of eclampsia, right? So you have 0 to 9. That's a major chunk where you have about 11,792 observations coming up. And you have a very small chunk of observations that end up having eclampsia to be a value of 1. And that's just 176 observations. So this is a pretty decent split in terms of eclampsia. But notice this, it has not used hypertension during pregnancy as a first split, although that was the most relative input when you had a look at the previous one under data exploration, right? So your decision trees would end up still using those relevant inputs itself. There could be a chance that your most relevant input ends up not being considered as a first split. If you come down, you then have this variable. You can go back to the data pane to check out. I'm sorry, I did not notice a label of this variable, so I did not know what was this variable. But then you have this second variable that's taking up the second most important split. And that's how you can read through the decision tree. You can also see the rules, the node scores in terms of your variable importance and the strategy in order to predict your outputs of 0 and 1. The tree, at what level did it get selected? What was your misclassification rate? And how many number of leaves are there? Leaves are your final categorizations that do not have subsequent branches coming out of it, right? So that's your final predictions in terms of a rule logic. Right, so that's something that you can look at. And following this, you can also get into the assessment tab. And the assessment tab is going to give you a lot of summary statistics in terms of your cumulative lift scores. You have gains, you've got capture response percentage, and so on and so forth. Now, you also have your ROC reports. Not that great. Here, you're just looking at a small number of values being captured at earlier deciles of your data. So I do not like this ROC. So, for obvious reasons, we would want to further train our model. I would rather get into I would rather want to do the more of the bivariate analysis by myself. And I would also want to manually select variables to be included in the model and then go ahead and maybe check and see how the decision tree then works. Right. So even if you go ahead and have a look at, the percentage plot or the count plot of the true positives or the true negatives and so on and so forth, you notice for this corresponding model, it's not that great, right? So that's a way you can assess your models, you can build your models, then, you can fine tune. So with the default options that I have for the decision tree, I don't get that great of a model. I can then also go ahead and change my different options. All right. So this is an example when you talk about uh, a point and click option in terms of just doing predictive modeling, machine learning, or any of the newer complicated models. Any questions, doubts still here? Welsh test if the Levines fail. So I'm not quite sure about the Welsh test. I'm sorry about that. That's something that I've not learned. I've always learned the Wilcoxon or the Kurzweil-Wallace. If anyone else is there in the group who is familiar with the Welsh test, then maybe you all can help Sunny with his question. Uh, Bottom line being, in case if the Welsh test is a non-parametric test, then yes, you can. If it's not a non-parametric test, then you would not want to use it. So bottom line being is that if your Levene's test fails, it means your parametric tests have failed. It means you would want to go ahead with a non-parametric test of analysis. And the non-parametric test of analysis that I have personally always dealt with are your Kurzweil-Wallace and the Wilcoxon. Those are more of the common ones that we tend to use. I have personally not used a Welsh test, so I would not be the right person to comment on this. So anyone else in the group who has used a Welsh test can please help Sunny with his question. Sorry, Sunny, for that. There's not much that I can help you there. All right. So just one last example that we are going to look at is relating to text analytics, which is like, the bottom um, starting point for large language models that you have in generative AI. So given that a lot of people do talk about generative AI these days, I thought that it is good to show that yes, we do have a very visual interface in order to work with text analytics. And again, your backend is the LITI. Uh, that's a baseline for natural language processing. And then subsequently, you also have large language models in the backend. And the use case that we're taking here is to look at anxiety and depression, right? So just a quick look, since we are running slightly behind time, I have again I just want to show you how the outputs come. So we've got a lot of user feedback information in terms of consuming certain medications for anxiety and depression, right? So some of the feedbacks that have been captured are in terms of the drug name. So you have Abidal as one of the drugs, and you have another drug known as Extapin that has been used for anxiety and depression, or to control anxiety or to control depression. And then following that, there are varying dosages that our patients have taken. And on the basis of their consumption of these medications with the dosages, they have given a feedback or they have given a response in terms of whether they do face any side effects or they're not facing any side effects, whether it has controlled their anxiety attacks or whether it has controlled the depression problems. So that's something that we have looked at. So all of that information has been captured across different drug reports, and they have all been combined into one single table. So the starting point of any text analysis is you would always want to tell the system what is it that you want to identify, right? So even when you use generative AI or systems like a chat GPT, you would always ask a question and then chat GPT is going to identify information in the backend and give you the relevant results, correct? So similarly, when you want to just overall go ahead and look at your drug reports, you would want to define certain concepts, right? So if I just go ahead, right click, and I say open, there are multiple different concepts that I have defined. Four very important concepts. First, like I said, we are looking at a lot of different medications, examples being Abital, then you've got Extapin, you have Concordin, Celifin, Captalan. So these are different medications that can be used in order to control the general anxiety disorder as well as depression. Following this, these medications are consumed in varying dosages, and the way you identify the dosages is with mg, because these are all tablets, they are not liquids, they are not vaccinations, right? So it is a tablet in milligrams. Then you have a prescription, so you can use certain, so there's an example of a regex, regular expressions, which is very generic again. So you can use regular expressions here, and you can also have certain classifiers, concepts that are very specific again to SAS, text analysis. Then you have the prescription. The prescription would be a combination of the medication and the dosage, so using two different concepts here. And then you have the list of the side effects. So again, all of these side effects are acting as classifiers. So on the basis of all these different concepts, you can try and identify how many patients have reported either having a side effect or having a specific dosage, or having a specific medicine with a prescription, right? So if I'm just selecting the side effects custom concept, now across my 1400 user feedbacks, so patient feedbacks, I can just get into the matched. And here I do notice it will start looking for all of these different terms in my documents. And then I do get to see that there are about 406 documents having the, or making a mention of a particular side effect, right? So the starting point of your natural language processing, or in order to work with unformatted textual information, would be very specifically to first try and identify the information that you need. On the basis of that, you create concepts. You can then club these concepts with each other. You can create a topic, and then you can plug in those topics into categories of very specific anxiety and depression. And that's something that the text analytics has as a default pipeline. So if you're looking at the entire process flow, keeping sentiment analysis away, if you're looking at the entire process flow, you start off by designing your concepts, what information you want to identify. Then you would want to identify different parts of your text, like what words have a commonly occurred during different feedbacks. A combination of your concepts and your text become your topics. And then you can club multiple topics to identify one particular use case, such as an anxiety or a depression. So that's something that you end up doing under categories. So if I just go ahead and show you the categories, then here there are three categories which I have created. One category which is relating to anxiety. So notice this, there are topics of anxiety and anxious which are coming into picture here. And then if I have one category very specifically relating to depression, and then I have a combined category of both depression and anxiety. So not necessary that my patients suffer from either of the two conditions. I can have patients suffering from both depression as well as anxiety. So if I go ahead and select the depression and the anxiety, I can get into the matched documents, and here I can see, sorry, my chair is again going down. So there are about 652 user feedbacks that make a mention of both depression and anxiety in the same feedback, right? So that's something you can do with the help of the visual text analysis. Okay. Okay, before I could come back to the last few questions, in case if you all have any questions relating to the predictive modeling use cases that we looked at, please feel free and go ahead and attach the questions in the chat window. In the interest of time, sorry, I'm just going to run ahead. So like I said, there are a lot of learning subscriptions available. Each of these learning subscriptions are going to look at a very specific set of knowledge that you can choose. They can also be mapped towards your job roles. Like, say, for example, if you do come with a biostatistics background, you do want to become a data analyst relating to healthcare and life sciences, then you have a biostatistics learning path. If you're interested to look at our SAS-based solutions, such as the life sciences analytics framework, health analytics framework, cohort builder. So like we said, over the analytics lifecycle workflow, you have the solutions and capabilities which stay on top of it, and that makes your end-to-end solution for the industry. So these are the different solutions that are there that are specific to healthcare. And if you would want to gain information on that, then you have the health and life sciences track for it. Then you have social and behavioral research. Following that, you've got Internet of Things. Internet of Things, like I said earlier, uses a tool or an application known as event stream processing that can work with queue-based data like RabbitMQ, Kafka, and so on and so forth. And what we looked at in terms of the entire analytics lifecycle, that is something that you can find under the SAS predictive analytics and machine learning learning path. Right. So each of these links, you can access them. I'm just going to go ahead and show them to you. We are just going to talk a little bit in detail here. So for those of you all who are looking at biostatistics, then you start off with the parametric tests and the non-parametric tests of analysis. Following that, you are getting into regression modeling. You're getting into causal inferences like propensity score models, Heckman's two-stage models, interrupted time series. These are something that's very specific to, biostatistics. Poisson regressions, again, because death as a count or major illness as a count is going to be a very small number. So, Poisson regression works. Then you have a lot of mixed model analysis. Then following this, you also have SAS programming for R users. If you do come with an R background, how you can go ahead and marry R and SAS. So that's also interesting. You then, again, since the whole world's talking about generative AI, you also have responsible innovation and trustworthy AI and how does it work under healthcare and life sciences. Right, so that's the biostatistics learning path. If you get into health and life sciences, here you're looking at all the solutions. So we have multiple different solutions like health analytics framework, cohort builders. Cohort builders are used for patient sampling, for clinical trials. Then you have your clinical enrollment simulator, which is again going to talk about randomization, sample size design, and so on and so forth. Following this, you then have the social and behavioral research. Now, again, since we have taken up certain examples relating to multi-generational studies, longitudinal studies, this becomes a very interesting topic or a learning path to look at. And this social and behavioral research has multi-level modeling for hierarchical and longitudinal data. Then you are looking at probability surveys. Then you're looking at structural equation modeling. These are something which are very complex and high-end statistical modeling that might not be relevant to other industries, but they are needed if you have to become a hardcore data analyst or data scientist for healthcare and life sciences. Then again, in case if you are one of those participants who would want to focus more on just the data management aspects, then integrating your REST API services, this becomes a very interesting topic for you. This is purely technical with Python and R. It just shows you how your REST API services work, how the integration with open source, how that marrying of the open source with the SAS interface is seamless. And then again, for obvious reasons, you have the responsible innovation and trustworthy AI, even coming up under social and behavioral research. You then have the Internet of Things. Internet of Things is purely relating to event stream processing. And following that, you are looking at the advanced analytics for IoT using event stream processing. So this is also going to show you how your streaming data can be analyzed or predicted on the basis of a model that possibly you would have already designed. It does have energy forecasting solutions here. This might not be directly relatable to you, but indirectly, again, it becomes a very important point because energy, utilities, healthcare, life sciences, these are all cross functional industries. And last but not the least, this is something that we looked at as a part of the entire analytics life cycle, the SAS predictive analytics and machine learning subscription, right? Now, there were a couple of questions. So all these links will be available in the deck file that I'm going to share a couple of minutes from now. And sorry, some of you all were asking the certifications. How do you go about with the certifications? Do you have practice exams? Do you have the certification preparation guide? So all that you need to do is get on to your country's SAS.com website. Now you can choose to use the US website, or you can choose to get into your region specific website as well. Either of the two are perfectly fine. Please excuse me. Yeah, so within the SAS.com website, I am on our headquarters website, which is a US website. Since this is a open session and we've got participants from different countries, hence I am not focusing on one specific country page. So under the Learn menu, you should be able to find certification. And here you have choose a credential, right? So this is a starting point. I'm going to plug in this link in the chat window because this link is not there in the deck file. Now, sorry again. Now, when I click on this choose a credential option, I have different tracks available with me, right? So when all that we looked at today is a part of programming, advanced analytics, visual analytics, and data management. We have not spoken about administration. We have not spoken about ideas. But for each of these different tracks, like programming, you can get into the certification, you can get further information of it. You can see what are the prerequisites, what are the previous credentials that you need to have, and so on and so forth. So one of you had asked a question relating to the business analyst certification. Yeah, the SAS certified statistical business analyst using SAS9 regression and modeling. So if I go ahead and open this in a new link, here you do notice that there are three options. You can either look at predictive modeler, you can look at visual business analyst, or you can become a machine learning specialist. This exam, what are the different questions that you would end up getting? You would get the exam details. In case if you are looking for training or e-learning content, then what corresponding courses you should be signing up for, or you should have knowledge equivalent to them. And under the additional training tools, you will definitely find the free practice exam. So this free practice exam, in order for you to access it, you can access it multiple number of times. In order for you to access it, you just need to have your profile being created on the SAS website, and it will take you to an environment known as a virtual learning environment. Right, so that's something that you can have a look at. So for some of the participants who are very specifically asked for this certification, as an example, I'm plugging this in the chat window. All right, now I am going to take up a couple of questions here. So Revati had asked a question. Do we have work from home job options with this SAS certifications? Now, that's a very hard to answer question. It kind of depends which corresponding company you get a job in and what model of work they have. Now, me being from SAS, I have a hybrid model. I either work from the office or I have a traveling job, in fact, because we have to go for customer training. So that's obviously on site. And then sometimes like this webinar, which right now I'm conducting it from the office, I can also conduct it from home. But it's going to be very company specific. So the answer to this question is yes, no, maybe. It all depends upon which company you get a job in, right? So that's the first thing. Next question is from Merdad. I'm extremely sorry in case if I'm going to pronounce your name incorrectly. Are all these datasets available on SAS via for learners? Yes, your understanding is perfect. And one of the reasons why we ended up using all the data that you have been looking at today, most of them, especially the predictive modeling ones, are definitely available on via for learners. All right, Tristan, what is the best way to ensure we get a copy of the recording and PDFs? So the copy of the recording will definitely be shared with you on email. The PDF, I'm going to go ahead and attach it right now in the chat window. So I request all of you to please go ahead and download the PDF that has been used for our presentation today. Last question from Sunny. After doing SAS certification, is there any possibility to join as a statistical programmer as a fresher? Oh, yes, definitely. Yep. There's no stopping you there. If your knowledge is good and if you're able to crack an interview, why not? Yep. So, in fact, ever since I completed my graduation, as an example, ever since I completed my graduation, I have been working into analytics, irrespective of SAS or otherwise. So I have around about 7 to 8 years of experience as on date. And it's been a combination of in-house analytics, it's been a combination of SAS, and it's also been a combination of a little bit of Microsoft-based analytical products as well. So if you have studied and if your background is relating to statistics, like I come with an economics and a statistics bachelor's degree, followed by more of a health informatics degree, master's degree. So I've never worked in any other area apart from analytics ever since I started working, right? So, yep, definitely, you will definitely get options as a fresher. You just have to crack the interviews. That's about it. All right, so a couple of more things that I wanted to show you all in the deck file that has been shared across to you. You would be able to see the very last slide, which is the contact person of our education team across the Asia Pacific countries. So in case if any of y'all do have further doubts or questions, in case if I have not taken up anything, please feel free to reach out to them. They can help you out. You want to sign up for any courses, you want to get trained or your team wants to get trained, we are there to help. Nonetheless, you can also email me. I would be going ahead and sharing my email ID in the chat window, which is ashwini.reddy@sas.com. So please feel free to write to me. In case if I cannot answer your question, I will definitely try and get the appropriate person within our team, within Asia Pacific, to reach out to you. While you all can keep the questions coming, since we are less than 1 minute away to 12:30, I'm just going to initiate a poll in order to get a feedback from you for this webinar. So in case if you don't have any doubts, don't have any questions, you can go ahead and just give us a feedback. And then feel free to drop from the session. You've been wonderful. And thanks a lot for joining in today. If you do have a doubts or questions, I'm just leaving the session on. You can go ahead and ask your questions. So I have just launched the feedback. So please go ahead. It's just a feedback of about 6 questions. Request all of you to go ahead and just plug in the feedback, and then you can go ahead and drop from the session for today. And if you have any doubts or questions, please keep the questions running. Request everyone to please give in the feedback before you all go ahead and drop from the session. In case if you are dropping, thank you for joining in, and hope I have been able to give you the right amount of knowledge in the short time of 4 hours. Thank you. And do reach out if you all need help relating to picking up knowledge. Thank you.